In the Carboniferous period, the beginnings of this new marine territory were already taking shape. By moving closer together, by succeeding, the land masses enclosed part of the Panthalassa in the hollow of their arms. They encircled this small piece of Panthalassa Ocean, which then became Tethys. Over the millennia, and as continents have moved, Tethys's place on our planet has changed considerably, but traces of its presence can still be found today. It's the Mediterranean Sea, but also mountains. That's right. Tethys was made up of shoals. These shoals, incredible as it may seem, have become the roofs of the world. Snow-capped peaks from the Himalayas in Asia to the Alps in Europe. We're going to land on the shores of this ocean and find out what's so special about it. Apart from this little anecdote about the remnants of Tethys's past, also known as Paleothys, this ocean is really a very special Permian body of salt water. Because of its position and encirclement, it became a veritable marine breeding ground. Life exploded and diversified within it. Thousands of species hatch in this blue jewel box. But why? Why here more than anywhere else? The Tethys Ocean is considered a veritable marine breeding ground. In some respects, it's as if we were in the Cambrian, a pivotal period during which life never ceased to enrich, develop, and diversify. Thousands of species found, all the conditions they needed here. I'd now like to ask you a question. Why do you think the Paleothys seabed is more conducive to this important diversification of marine species than the rest of the vast Panthalassa? What changes the situation? Closure by land is not the only reason. The answer lies at the top of these mountains. In fact, it's the erosion of these great mountain ranges that carries so much sediment into the ocean. These sediments are the preferred source of nutrients for aquatic plants and marine microorganisms. The base of the food chain is enriched by a new nutrient source, and the whole chain benefits. Let's start by taking the chain back to its base. Fusilians appear on the seabed. These fusilians are small marine organisms. These little grains of wheat are the famous fusilians. They are unicellular organisms. In other words, a single, highly complex cell performs all the basic functions of life. Fusilians secrete calcium carbonate from the protoplasm, the cell's living material, to form the hard shell and its various chambers. Other small organisms like this will enrich the seabed, creating a new ecological niche. Corals also spread widely, enriching the ecosystem by providing shelter for these multiple marine species. There were two main groups in the Permian, Tabulata and Rugosa. Their calcite exoskeletons favored the diversification of other marine species. Sponges, equally adept at architecture, also form numerous reefs. Unlike corals, which are built from calcium carbonate, sponges are made of silica. They are colonial and, above all, filter feeders. Some species are very large, like Gigantospongia, which can reach 2 meters or 7 feet. Gyrtiocelia forms long branches like corals but is really a sponge. Brachiopods, but also cephalopods, goniotites, and nautilus will once again diversify and enrich their genus with new species. Here we can see a nautiloid, Cuperoceras. As in the Carboniferous, brachiopods and echinoderms carpet the seabed, but look how numerous they are. They come in all shapes, sizes, and colors. Here are Leptidus brachiopods, and next to them, Edria stegus. Further up the reef, Spirifer brachiopods. Various marine plants, microorganisms, and the famous sediments from the mountains help all these species to expand, with mollusks, brachiopods, crustaceans, and even sponges and corals benefiting from these new living conditions. The base of the food chain is therefore rich, diversified, and abundant. With the abundance of food available, the species that feed on them also proliferate. Let's move on up the water column. We find gonionites, but they're not the only ones swimming in this water column. Have you noticed how many fish there are here? Just ahead of us is a whole shoal of Platysomus. A little further on, Dorypterus. The fish live here by the thousands. When prey is so plentiful, predators respond. They act as regulators. It's one of the oldest laws of nature. With so much choice on the menu, predators have also diversified. Some are large, others very powerful. Let's discover them without further ado. Do you remember who was at the top of the marine food chain in the Carboniferous period? Yes, sharks. Or to be precise, chondrichthyans, a cousin of today's sharks. They became the kings of the ocean when, at the end of the Devonian period, the placoderms disappeared. That didn't change much during the Permian. Quite the contrary, in fact. They continued their ascent to establish themselves as major marine predators. Hybotus conquered both fresh and saltwater. This member of the Hypodontes reigns supreme over the aquatic world. It looks a little less like the sharks we have come to expect, but they too are chondrichthyans alongside Hybotus. These are the xenocanths. Xenocanthus has a ribbon-like dorsal fin that runs the length of its back. You may have noticed its large spine pointing backwards from the head. Scientists are still struggling to determine the purpose of this famous spine. Some think it may have been venomous, making it easier to hunt the crustaceans and fish it feeds on. In a completely different vein, I'd like to introduce you to Genassa bituminosa. This is a pedilondonte. It has a very distinctive shape. It looks a bit like our modern-day stingrays. Genassa bituminosa feeds on all the marine shells we encountered earlier, in particular, the many brachiopods that abound on the ocean floor. Finally, I'd also like to tell you about the enigmatic Helicoprion. It too feeds on these mollusks, crustaceans, and brachiopods. But to do so, he has a rather unusual weapon. Helicoprion is a eugenodon fish. Although it looks a little like a shark, it's not. It measures up to 10 meters or 33 feet. Its dental whorls reach up to 40 centimeters or 15 inches in diameter. This spiral dentition is truly astonishing. It is undoubtedly an adaptation for feeding on soft-bodied prey and may have functioned as a shelling mechanism for hard-bodied cephalopods such as nautiloids and ammonoids. In a word, it's a real shell crusher. Perhaps it's time to go up to the surface and see what's going on in plants and vertebrates. There have been many changes. Carboniferous plants were largely spore plants. In other words, they needed water to develop and maximize their chances of reproduction. A few seed plants have begun to appear, but their presence is still timid on the continent. Most of them reproduce by sporangia. This is the case of Sigillaria and Lepidodendron, which were very common during the Carboniferous system. As we have seen, swamps are drying up in some regions. The land is gradually becoming an oven. If the soil gets drier and drier, water can no longer serve as a channel for spore-forming plants. What's more, these plants acclimatize to a humid rather than tropical environment, rather than to arid soil and dry air, not to mention reproduction, find it increasingly difficult to find sufficient energy to grow and develop. The forest of these arborescent plants is dying a slow death. These new, extremely difficult conditions have taken their toll. They're disappearing. On the other hand, one group of plants managed to survive in this seemingly hostile environment during the Permian. These are the bare-seeded plants known as gymnosperms. Remember their timid appearance in the Carboniferous? Well, the least we can say is that in the Permian, they became much more widespread on the continent. These include, but are not limited to, the famous seed ferns. Let's take a closer look. At the end of the Carboniferous period, they gradually began to colonize the Earth's soil. This was only the beginning of their colonization, of course, as spore-bearing plants were already on conquered ground, so it was difficult for them to find a place for themselves in this dense, lush plant cover. But when drought came knocking on the door of this entirely water-dependent forest, spores were forced to give way. The conifers will take advantage of the vacant space and take over the land, but not just anywhere. As you know, the climate has undergone many changes over the course of the Earth's history. Depending on where you are on the globe, you won't find the same temperatures. On either side of the mountains, you can find rather humid zones or arid lands. There are also more temperate zones with clearly defined seasons. The conifer family will not develop in the same way everywhere, depending on the region. The Walchia conifer, for example, establishes itself in the north. Walchia piniformis is a conifer somewhere between Cyprus and Araucaria, a large conifer in the Andes. The cone-shaped fruits are the source of this family's distinctive name. To the south, we find the Glossopteridales. These trees can reach 30 meters or 100 feet in height. The wood of their trunks and branches has rings, similar to those of some of our contemporary species. These rings are invaluable witnesses, as they enable scientists to confirm one thing. In these temperate zones, the climate has clear-cut seasons. To adapt to these sometimes extreme changes, these trees have adopted a new strategy: deciduous leaves. At the same time, cycads also appeared. They are considered to be close to conifers despite their deceptive appearance as a cross between a fern and a palm. The reason they are so close to conifers is that they too contain the various reproductive organs which are located at the heart of the leaf crown in the form of a modified leaf cone known as a strobile. The cycadophytes, follicularis, and Cycadophytes granulatus, found here, are only just beginning to colonize the soil. In the Mesozoic, the Cycadales group expanded. This great diversification of trees and zonation of plants enabled new ecological niches to emerge. New evolutionary radiations are underway. As we saw at the end of the Carboniferous period, amphibians gave rise to amniotes, animals capable of laying eggs with shells. Without this ability, these amphibians were completely dependent on the aquatic environment. Indeed, amphibians had legs to move around and lungs to breathe. All they lacked was the assurance of being able to protect their offspring on land. Until now, amphibians had to lay their eggs in water. To free themselves completely from this element, they had to update their mode of reproduction. The amniote class has the ability to lay eggs with a shell and amniotic fluid inside. The amniotic egg enables embryos to develop serenely on the Earth's surface. It can withstand air pressure and any microshocks on Earth. But evolution didn't stop there. These amniotes were to define the major evolutionary lines of the Permian. They define the new face of fauna in the era that closed the Paleozoic. They gave rise to new clades, the sauropsids and synapsids. We'll come back to the latter in a moment. These two branches are undoubtedly descended from two species that we had the opportunity to crossbreed during the Carboniferous. At the very end of the Carboniferous, to be precise. For the sauropsid branch, it's Hylonomus laeli. For the synapsid branch, it's Archaeopteris florensis. Between them, they gave rise to many of the terrestrial species that populated the continent in the Permian. Over the millions of years of the late Carboniferous and the three periods that define the Permian, species evolved and diversified. The path of this evolution, and what it will lead to, is very interesting to observe. Before discussing the emergence of synapsids and sauropsids, it's important to understand and differentiate certain technical terms that define their characteristics. Those of particular interest to us are those used to classify amniote skulls according to the number and position of temporal windows: anapsid, synapsid, euryapsid, and diapsid. When several species are subjected to the same environmental constraints and ecological niches, they will independently adopt several similar physiological, morphological, and sometimes even behavioral traits. This is known as convergent evolution. The different temporal windows mentioned earlier are part of this type of evolutionary convergence. Depending on their environment, but also their ecological niche and therefore their diet, each of these species has evolved according to its own needs. The temporal window houses the temporal masticatory muscles. The strength and position of these muscles respond to a particular need, depending on the diet and type of prey, for example. The contribution made by the different bones to the edges of these temporal windows varies from group to group. When we speak of anapsids or parareptiles, as the term anapsid is controversial among some scientists, we are referring to a group of amniotes that have no temporal fossa. Synapsids are reptiles with skulls pierced by a single, lower temporal window. Finally, diapsids have two. The diapsid skull is characterized by the presence of upper and lower temporal fossae. According to recent research, these same diapsids, despite their two pits, may have become euryapsids or anapsids. Both may have lost one of the pits during their evolution. The term euryapsid is also controversial, as its meaning is ambiguous. It can refer to all amniotes with an upper temporal window, or only to direct descendants of the first diapsids. The euryapsid form corresponds to the presence of upper temporal fossae only. Now that the shape of their skulls holds no secrets for you, let's take a closer look at their morphology, ecological niche, and various characteristics. Pelycosaurs are many and varied. They come in all shapes and sizes. Pelycosaurs include several families, such as Cassidae, Varanopatae, Ophiocodontidae, Edaphosauridae, Eotherididae, and Sphenocodontidae. What do they have in common? They all belong to the mammalian branch. We call them mammalian reptiles, although today we prefer the term pre-mammalian. Among them is the famous Dimetrodon, Sphenocodontidae. So here we are with our very first synapsid. It's quite something. Perhaps given its morphology and appearance, you're thinking that we forgot to specify one thing, the emergence of the first dinosaurs. But no, we haven't forgotten anything. Dimetrodon isn't a dinosaur. It's a reptile. The big difference between the two is that all dinosaurs, or sauropsids, have two pairs of temporal fossae, whereas Dimetrodon has only one. Dinosaurs will appear 40 million years later, so we'll have to be patient. Dimetrodon is therefore a synapsid, not a sauropsid. It's also a quadruped with a very special feature, its dorsal sail. It's impossible to confuse or miss it in the middle of this sparse wetland plain with such a fan on its back. The usefulness of this sail is still a matter of debate. Many scientists have studied the subject and are still trying to unravel the mystery of this singular physical attraction. It may be that the sail plays a thermoregulatory role, evacuating heat or, on the contrary, capturing it to warm the rest of the body. Others, however, suggest that it's more a means of courtship to seduce the opposite sex. Unless, according to the latter, it's simply a useful means of stabilizing the skeleton, and in particular the spine. Let's take a look at the rest of his skeleton and morphology. Its skull is high and curved, with large teeth of varying sizes set along the length of the jaws. There's no doubt about its diet. It's a perfect carnivore. Its favorite prey is fish, but it's not averse to a small tetrapod, a reptile, or even an amphibian. It shares its ecosystem with animals such as Archaea, Diplocaulus, Eriops, Captorhinus, and Edaphosaurus. Dimetrodon is a synapsid belonging to the sphenocodont clade. Not all sphenocodonts have a veil like Dimetrodon. Haptidus, for example, also belongs to the sphenocodont clade. It looks more like a huge chameleon. Also a quadruped, its tail is as long as its body. Now here is an Edaphosaurus from the Edaphosauridae family. This time we're dealing with a herbivore. We should even call it a vegetivore to avoid any confusion. We often speak of herbivores as opposed to meat eaters, but herbivore means eating grass. Grass was not present in the Permian. There is a multitude of plants, of course, as we have seen and will soon see again on this trip, and vegetation has greatly developed and even evolved, but for the moment it's still just plants of various shapes and sizes, not grass as such. Our Edaphosaurus, then, is a vegetarian, feeding on the plants it finds along the way. It is one of the oldest known herbivorous tetrapods. It measures between 1 meter or 3 feet and 3 meters or 7 feet, and weighs around 300 kilograms or 660 pounds. You'll notice that it too has a lovely dorsal sail. We could go on with our synapsid investigations and describe a few other species, but the most important thing to remember about this group is its evolution. The synapsids will give rise to the therapsid branch. In a few minutes you'll understand the importance of this group. In addition to the large Pelycosaur family, the synapsids gave rise to the therapsid family. These rapidly evolved and diversified. Just a few million years after the appearance of the first therapsids, a new branch of vertebrates was born. Let's take a look at these morphological evolutions by studying different species of Permian therapsids. The first therapsid I'd like to introduce you to belongs to the order Dinocephales. It's even a suborder of Tepinocephalus. This is Moschops. Yes, it's rather huge. It's almost 3 meters or 10 feet long and must weigh close to a ton. This semi-aquatic giant is an imposing plant eater. Knowing that plants don't provide much in the way of calories and energy, and given its huge dimensions, we estimate that it would need to eat over 100 kilograms or 220 pounds of leaves a day to meet its needs. As I was saying, it's semi-aquatic. By this I mean that Moschops lives a bit like our modern-day hippopotamuses. He is able to go on the ground but thrives in bodies of water. If its size is relatively exceptional, it's above all its gait that I'd like to highlight here. Until now, amphibians and parareptiles had a very arched, crawling gait, like many of today's reptiles. Here we're closer to a mammalian gait. The elbow joint, for example, offers a wider opening. The gait is therefore quite different. Another therapsid, much smaller but just as interesting to observe, is Lycanops. It measures around 1 meter and probably weighs no more than 50 kilograms or 33 pounds, but this doesn't stop it from being a very good hunter. He hunts prey within his reach, of modest size, such as a few sauropsids that we'll no doubt meet a little later, but also some members of his clade, small therapsids, notably diconodonts. Lycanops is an agile predator. Once again, I'd like to take a close look at its mode of movement. Like Moschops, it is far from having an arched, crawling gait typical of all the quadruped vertebrates we had encountered before them. In fact, it's quicker in its movements than anything we've seen before. Thanks to its long, close-to-the-body legs, it is more stable, and its movements seem easier, like quadruped mammals. Its mouth is a formidable weapon. It has a long, slender head, like the Canidae genus, but above all, it has fang-like teeth on both the upper and lower jaws. They act like daggers, they kill and tear. The flesh of this prey won't resist such perfectly sharp blades for long. In its mouth, it's probably a Robertia. It had the misfortune to fall under the fangs of the Lycanops. It does, however, provide an opportunity to evoke a new family of therapsids. Robertia is a therapsid that belongs to the Diconodont family. In fact, it was one of the very first of its kind in the Middle Permian. It is a sort of primitive diconodont in the sense that it is one of the oldest in the branch. This little plant eater didn't stand a chance against Lycanops. The small canine teeth protruding from its upper jaw are only useful for cutting through the toughest stems. Once free, it can sever the stem with its horny beak and enjoy a well-deserved meal. Another member of the Diconodont group we'll be looking at now is more imposing, measuring over 1.20 meters or 4 feet on average. Also a vegetarian, it is not immune to carnivores despite its more respectable size. Predators are numerous here, powerful and ferocious. Diconodon has to be on his guard. As it cuts vegetation with its beak or uproots tubers and roots with its tusks, danger lurks. Before closing this part on the therapsids, I'd like to turn to the Upper Permian with another family, the cynodonts. Procynosuchus is a semi-aquatic cynodont, as was Moschops. Measuring around 60 centimeters or 25 inches long, it appears to have the body of a crocodile and the head of a dog. It has a long tail, flat feet, and a long rounded head. In the water, it is perfectly at ease. Like an eel, it wriggles to swim. Its tail acts as a propeller in the water and is a great asset for gaining speed. But it's not all about the tail. Its legs are also very efficient in their own way. Its flat feet become paddles, and thanks to flexor muscles that can be identified by the ridges on the femur, it can bend and unfold its legs to increase thrust through the water. So that's all we can say about the therapsids. By now, you're probably wondering what's so important about therapsids. Well, when we introduced this group, I mentioned that this lineage was very important for the further evolution of living organisms. We didn't quite get to the bottom of it, but I did drop a few hints to put you on the right track, especially when we looked at the gait of certain animals, for example. Do you remember Lycanops? The movement was no longer reptilian, but tended more towards that of mammals. Have you guessed the key evolutionary step that led to therapsids? Therapsids give rise to a new branch, the mammals. These, in turn, gave rise to the mammals, a group that today occupies an important place on our planet. But it all began here in the Permian with the therapsids. In this second part on therapsids, rather than approaching these animals by family, we're going to discover them by period. In this way, you'll be able to see that in the lineage of the therapsids, the rapid evolution of these animals enabled the first morphological characteristics of mammals to be outlined in just a few thousand years. As its name suggests, Sphenacodon is a member of the Sphenacodontidae family, just like Dimetrodon or Haptidus, which introduced our synapsid group. We're in the lower Permian here. We're a long way from the characteristics you'd expect from a mammal. It adopts a much more reptilian morphology. It resembles a lizard, or more broadly, a reptile. It appeared in the upper Carboniferous and was still present in the lower Permian. At 3 meters long, it had very sharp teeth and was therefore a carnivore. Let's move on to the middle Permian. Let's start with Biarmosochia. The silhouette and head shape are already less reptilian. The head is smaller than that of the Sphenacodontidae presented earlier. However, the temporal cleft is larger than in the latter. Also noticeable is the greater flare at the back of the skull, where the muscles are attached. This means that the muscles are larger, resulting in a stronger bite. It is also equipped with enlarged canines. Canines, biting muscles, a smaller head that's better proportioned to the rest of the body. We can already see that evolution is underway, although Biarmasukia still retains some very primitive features. This is also true of the order Dinocephales. Their members include vegetarians, carnivores, and even omnivores. While some are exclusively terrestrial, others are semi-aquatic. Dinocephales have diversified greatly over the course of their presence on our planet. Although they are still very primitive, there have been some important changes. The ilium, for example, the upper part of the coxal bone, which along with the sacrum and coccyx enables movement of the pelvis, is now more elongated. Movement and gait are therefore more stable and comfortable. In carnivores, if we focus on the head, we can also see some changes, notably in the differentiation of teeth. Some at the front are longer and can shear. On the lower part, we can see that the teeth along the tongue interlock and are characterized by a crushing surface when the upper jaw meets the lower. Chewing can almost begin. Let's take a look at some therocephalons. Therocephalons are the closest relatives of the cynodonts that gave rise to mammals. A close look at the skull skeleton of one of them reveals the appearance of the secondary palate. It separates the nasal cavity from the oral cavity. This is a very common feature in mammals. Gorinicus is a therocephalus, like Gorinicus macuatinae and Gorinicus sundarensis. Depending on the species, the upper dentition consisted of 4 to 5 incisors, a large saber-toothed canine, and 3 to 4 postcanines. The articulated cervical vertebrae, the posture of the legs under the belly, and the pelvis favor a more stable upright gait. In other words, the gait is more mammal-like than reptiliform. It's already time to continue our evolutionary journey by discovering the species of the Upper Permian. Here are a few members of the Gorgonospians. One of them is an Inostransevia. It's the largest. It's close to Ursid size at 4 meters or 14 feet long and around 350 kilograms or 770 pounds. Its head is about 45 centimeters or 18 inches long, which is quite reasonable and proportional to the rest of the body. Its teeth, on the other hand, are very long, about 12 centimeters or 4 inches. This admirable length, however, is not so developed for all the teeth. Only the canines are. The others are specialized like ours, each with a particular shape depending on whether they cut, shear, or crush. They are gigantic and terribly frightening. However, if we put aside their monstrous appearance, their name comes from the Gorgons, the snake-haired women of Greek mythology. Then we can glimpse new, evolutionary stages. This mammalian reptile lived in the Siberian desert during the Permian era, one of the driest periods of the Paleozoic era. It adapted to these new conditions. Its legs, feet, and ear bones bring it closer to mammals than reptiles, even though it undoubtedly belongs to this group. Let's finish with the order that gave rise to the mammaliforms and soon to be mammals, the order of cynodonts. Let's take a closer look at Thrinaxodon. The teeth have evolved considerably and are now perfectly differentiated. The cranium is also enlarged at the back of the head. The zygomatic arch in the skull has also widened, bringing it closer to that of mammals and allowing for a more robust jaw musculature. But there's another important thing to discover about Thrinaxodon: the fur. At least that's what some scientists think. Thrinaxodon was undoubtedly an endothermic species. Before moving on to the second group, the sauropsids, which are also important in today's world, let's take a look at the two characteristics of cynodonts and diconodonts that scientists have recently discovered: dentition and thermoregulation. Decynodont means two dog teeth. This is an order of herbivores. In addition to its upper jaw tusks, the diconodont had a turtle-like beak, which it used to chew its food. The diconodont order was highly diversified, with members ranging in size from a rat to a large elephant. And speaking of elephants, many of today's animals have tusks, including elephants, walruses, and warthogs. These animals are all mammals. This was not necessarily the case at the time, however, as a new evolutionary process was clearly set in motion in these therapsids, which would later give rise to mammals. Tusks are defined by teeth that protrude from the mouth and grow continuously, as is the case with some diconodonts. In this case, the tusks are made of dentine, but this is not the case with all diconodonts, as some have no tusks but do have teeth. These teeth also have an interesting peculiarity to note about their composition. Here, it's not dentin, but hard enamel, a material also widely found in mammals. These are very large teeth, canines made of enamel. Dating analyses were carried out on 90 fossils as part of an international collaboration. These analyses brought to light a very important fact about two groups of therapsids, the diconodonts and the cynodonts. They were undoubtedly warm-blooded. Today, birds and mammals are the only animals capable not only of producing their own body heat, known as endothermy, but also of maintaining it at a constant high temperature, known as homeothermy. The combination of these two characteristics in mammals and birds is referred to as endohomeothermy. This invaluable characteristic, found in mammals today, is thought to have appeared in therapsids as early as the Upper Permian, between 252 and 259 million years ago. According to the study, 63 of the 90 fossils studied were therapsids of 22 different species, including diconodonts and cynodonts. The researchers analyzed what is known as the isotopic composition of oxygen. A chemical element can take on different forms, known as an isotope, with a nucleus of different mass, depending on the different isotopes. In other words, an isotope is a kind of fingerprint of a chemical element under given conditions, in this case in relation to oxygen. Stable oxygen isotopes are incorporated differently into bones and teeth, depending on the animal's metabolism. By analyzing this data, we can determine whether they are cold-blooded or warm-blooded. In this way, the group of scientists was able to determine new evolutionary perspectives in two of our therapsids, since cynodonts and diconodonts were probably warm-blooded and capable of endo-homeothermy. Are the two related? It's hard to say for sure. Even so, the majority of terrestrial species will disappear as a result of the mass extinction that is fast approaching, with the exception of diconodonts and cynodonts in particular. The key to their resistance to abrupt climate change may lie precisely in their endo-homeothermic capacity. These discoveries allow us to close the chapter on therapsids and the emergence of mammillomorphs. Over the course of evolution, these cynodont species gave rise to the mammaliforms, then in turn to the first mammals. So, with the origin of this group, our origins began millions of years ago in the Permian. Let's open another chapter with the sauropsids. Sauropsids are a clade of tetrapods and a sister group to the synapsids we've just seen. Together, they form the taxon Amniotes. Sauropsids are divided into several branches, including anapsids, which have no temporal fossae, and diapsids, which have two. Let's start with the parareptiles. These are diapsids that may well have lost their temporal fossae. Our very first reptile is not terrestrial but aquatic. To meet him, we'll have to leave the peacefulness of the forest to reach the large lake at our feet. It's a veritable fish pond here. Countless species of fish and other freshwater animals share the territory. Despite the apparent calm and the richness of the seabed covered in seaweed and other marine vegetation, danger is ever present. As we descend a little deeper into the water, we should cross paths with our famous Mesosaurus. And here it is. With its meter-long, lanky form and long tail, it's impossible to mistake it for any other. It belongs to the anapsid reptile genus. It was one of the first reptiles to return to the water after the amphibians had left it to settle and evolve on land. Its nostrils are located on the top of its skull, enabling it to breathe with only the upper part of its head protruding, rather like our modern-day crocodiles. Its morphology suggests that it moved by undulating its body, but it was also capable of diving to the depths. Its thoracic cage is strong enough to allow this in any case. His ribs are banana-shaped and very thick. This means they can withstand the pressure of the water. This ability is very useful for feeding on the seabed. It hunted small crustaceans and other small prey, which it captured with its jaws, equipped with numerous elongated, fine, pointed teeth. Despite its great adaptation to the aquatic environment, Mesosaurus may well have had to visit land during its lifetime, but only for one very special occasion, egg-laying. This would bring him closer to a way of life that we still see today in sea turtles. Like these turtles, the shape of its feet makes it very difficult to move around on land, and it is more at ease in the aquatic world. Like Perrhiasaurus, Mesosaurus belongs to the anapsid clade. Although both belong to the same class, they do not share the same ecological niche. This one is a terrestrial quadruped. So let's get out of these murky waters and onto dry land. This is Perrhisaurus nasicornis, one of the first of the Perrhisaurus genus. It's a large animal with a skull about 50 centimeters or 20 inches long. Its snout is fitted with a solid carapace. Perrhisaurus has many teeth, even on the palate. These are not useful for attacking meat, but rather plants. Its teeth are ideal for cutting hard plant fibers, as they are topped by cusps. Cusps are the sharp points at the top of the teeth's dentine. The Perrhisaurus family also includes Scutosaurus. Its name means shield reptile, in reference to its bony armor and the many spikes decorating its skull. It measures around 3 meters or 10 feet. As you can see, he's big, but also very heavy and imposing. However, his skeleton is not very well adapted to walking. It moves slowly and with little ease, so it's hardly surprising that more powerful reptiles such as Enostransevia or Gorgonopsian, whose path we've already crossed, easily overcame this large Scutosaurus. Its only defense was its muscular body and strong bones. Rows of hard bony plates under his skin covered his powerful muscles. But this didn't dampen the ardor of the most reckless or the hungriest, as is clearly the case with this Enostransevia. Scutosaurus must undoubtedly have known the hunger in his belly, because in the semi-desert region where he found himself, there was very little vegetation. He must therefore travel many kilometers, often in the open, and therefore at the mercy of predators. When the precious plant treasure is finally within reach of its mouth, it uses its flattened teeth to crush the leaves and get to the bottom of the young branches. To digest, it is one of the so-called lithophagous animals. To digest plants, they ingest gastroliths, i.e. stomach stones. Stored in the gizzard or stomach, they facilitate the animal's digestion. Not all Permian sauropsids were large animals. Look at this one. We could have passed it by without even noticing its presence. So well does it blend into the background. This Procolophon is a small parareptile measuring around 30 centimeters or 12 inches and weighing just 2 kilograms or 5 pounds. It's rather stocky, as you can see, and scientists believe that its short, powerful legs helped it to dig burrows. To feed or protect itself from predators, nothing is less certain. It has several characteristics that tend to place it in all possible categories: vegetarian, insectivorous, and perhaps simply omnivorous. While this one is quietly enjoying his meal, we'll be taking a look at another group of sauropsids. The skulls of these amniotes have 2 temporal fossae, so we're talking about diapsid types. In sauropsids, as in synapsids, a new branch appears. A branch that little by little will also lead us to contemporary animals. The diapsid branch will in fact lead to several distinct evolutions. Ongoing studies and research sometimes contradict previous results, meaning that the classification of these animals is still a matter of debate. The diversification and evolutionary process is so broad that it's not always easy to make sense of it all. Let's take a look at the findings of some researchers and paleobiologists. The sauropsids are the origin of several lineages, or branches, which we're going to study in a little more depth. The testudines, diapsids, and archosauromorphs have many secrets to reveal about the evolution of living things. Let's start with testudines. It's through their study that scientists have been able to understand one of the evolutionary stages of diapsids. To understand the lineage of turtles, and therefore of testudines, we need to go back to the probable origins of this group, and that leads us to Unatosaurus. Unatosaurus africanus is a kind of prototorture. Its classification is still a matter of debate, with some scientists thinking that it too could be part of the large Periasaur family we saw a few minutes ago. Recent research, however, tends to refute this hypothesis, to which we'll return after a brief presentation of Unatosaurus. Many physical characteristics suggest that Unatosaurus was at the origin of the turtle branch. His silhouette and morphology suggest it, but it's his skeleton that will help to support this hypothesis. It has broad, elongated ribs that curve around its back and belly. It has 9 elongated trunk vertebrae, 9 pairs of broad T-shaped dorsal ribs, and 5 pairs of gastralia, bony dermal plates also known as ventral plastrons. With such a skeleton, it's easy to guess a form of protoclea that would later become a carapace. Unatosaurus was a burrowing animal. The evolution of its skeleton may well have been a response to its burrowing needs. To facilitate burrowing, the ribs widened and flattened. But protection from predators can't be ruled out either. Indeed, today's turtles have preserved and even improved this physical aptitude, whether or not they are burrowers. The prospect that it could also be a weapon of defense against predators is therefore entirely plausible. Swallowing a round animal with such a carapace is very difficult. Let's see what scientists have discovered about Unatosaurus. A team of researchers from Yale University in the USA have uncovered an interesting point about the evolution of these animals. In juvenile Unatosaurus, they have identified small openings characteristic of diapsid reptiles. In other words, 2 temporal fossae. Later, however, they closed. Unatosaurus became an anapsid. This means that anapsid testudines may have evolved from diapsid reptiles during the Permian period. If this is the case, then we can rule out the proposed origin of Periasaurus mentioned earlier. While turtles, the order of testudines, were previously considered to belong to the subclass Anaspidae, today some believe that testudines are diapsids that have lost their temporal fossae. For the moment, the question is still being debated by scientists, but these results favor the hypothesis that diapsids gave rise to anapsids. Speaking of diapsids, it's about time we met a few of them, don't you think? Diapsids are a sub-branch of sauropsids. While anapsids have no temporal fossae, diapsids have 2. Diapsids have a plurality of environments and niches. While many of the previous sauropsids live on land, the next one prefers large expanses of fresh water. Hovasaurus is much smaller than our earlier Mesosaurus, but he has much to teach us. He is just 50 centimeters or 20 inches long. Its tail alone accounts for half its total length. Its tail is flattened on both sides, probably enabling it to swim by undulating its body. The lateral undulations of the tail could in fact provide sufficient propulsive thrust to move the whole body. In a way, it resembles a lanky marine iguana. Surprisingly, paleontologists have also found stones in its stomach. This is quite common in vegetarians to aid digestion. But Hovosaurus is not a herbivore. It is a piscivorous fish-eater. Stones like these therefore raise further questions. It may well be that despite this diet, Hovosaurus used gastroliths to aid digestion. But for some scientists, another answer is possible. They were useful for diving. He would swallow these stones to dive faster when hunting. Gastroliths are found near the abdominal cavity, a soft tissue membrane specially designed to contain them. Given the location of these stones, this means that Hovosaurus's center of gravity is at the rear. In these circumstances, a rearward placed center of gravity would undoubtedly have increased forward thrust through the sinuous movement of the tail, enabling it to dive more easily. Hovosaurus was therefore an animal that could reach a certain depth. The way it swam and its diet are among the clues that enable researchers to advance this hypothesis. But they are not the only ones. Another fact also supports this hypothesis. This time it's its skeleton. The study of fossils reveals pachyostosis in this animal. This is a bone specialization often found in amniotes secondarily adapted to aquatic environments. The bones undergo a thickening, generally caused by additional layers of lamellar bone, which increases the animal's weight, like a kind of ballast. This increases speed and depth, as well as swimming ability. This diapsid, therefore, develops more and more skills to adapt to new living conditions. Let's continue our investigations with another family. If there's one diapsid family not to be missed, it's the Archosauromorph family. As we've seen, sauropsids gave rise to diapsids. The latter have left their mark on Earth's history, and some of their descendants are still part of our lives today. In fact, among these diapsids, we see the appearance of those grouped together in the Permian under the name of Archosauromorphs. They form one of the keys to the evolution of contemporary species. After the Archosauromorphs came the Archosauriforms and Archosaurians, from which came dinosaurs, flying reptiles, and even birds. Once again, many of our contemporary species originated here in the Permian. Let's hear what these Archosauromorphs have to say. Protorosaurus is the oldest of its kind known to scientists. He's a bit special, I must admit. This insectivore does indeed resemble a lizard, but with long legs and a disproportionately long neck. This morphology, surprising as it is, seems to be more of an asset than anything else. Given its musculature and legs, it's safe to assume that we're dealing here with a fast-moving reptile. Towards the end of the Permian, these Archosauromorphs gave rise to the Archosauriforms. This category includes Proterosaurus fergusi. It was a predator and undoubtedly one of the largest terrestrial reptiles of its time. It measures almost 3 meters or 10 feet. Its long legs are perfectly designed for running. When it's chasing prey, it looks even more formidable. This herd of Lystrosaurus gently crossing the stream should be on its guard. This small herbivore is a burrowing animal of the order Synapsidae. It is a diconodont. Its stocky body, sturdy limbs, and famous pair of tusks on its upper jaw will be far from enough to cope with the mighty Proterosuchus lying in ambush. It feeds on fish, reptiles, and other animals of varying size that come to drink at the waterhole. He occupies this watering hole patiently, waiting for an opportunity to present itself. Like the crocodile, ambush defines its hunting strategy. Proterosuchus shares other characteristics with the crocodile, although there are also many differences. Like crocodiles, Proterosuchus has powerful jaws with conical teeth. Neck muscles complement its armada of sharp teeth, enabling it to open and close its mouth very quickly. He is impressive, even from a distance. It also has short muscular legs, but unlike crocodiles, they offer greater ease on the ground. So perhaps to a lesser extent, it also hunted on the ground. What crocodiles don't have either, unlike our Proterosuchus, is a hook-shaped, downward-pointing premaxilla on its upper jaw, which is very useful for slippery prey. It also has a few trump cards to play if it and its descendants, or at least the various evolutions that will follow, are to survive. It is able to cope with extreme situations. For example, some researchers believe that it stores fat in its tail and can go for months without eating. When prey is in short supply, it can hold out for long periods. It is also able to regulate its body temperature by alternating between sunbathing and bathing in cool water. Here again, given that the Permian continents were in a completely different position, with its habitat closer to the South Pole, it was able to acclimatize more easily to the changing seasons. Our Archosauriform thus opened the door to future Archosaurians and later dinosaurs, crocodiles, and birds. While future Archosaurians prepare to make their grand entrance on the scene, other reptiles have also adopted interesting new abilities. Before parading through the air, reptiles have gone through several intermediate phases. Predators on the ground are increasingly ferocious and cut out to attack any species. Pelycosaurs, for example, are widely present on the continent. Smaller reptiles don't stand a chance, unless they manage to conquer another environment. And that's exactly what some will do. A new environment is the only solution. So they've become arboreal. They live in trees, particularly in the canopy. This zone is richer in light, but also in insects of all kinds. In other words, there's an abundance of food to feed these new arrivals. Meet Coelurosaurus. It has an elongated, flattened body. It measures around 40 centimeters or 16 inches and blends easily into this landscape in the heart of the canopy. Coelurosaurus's special feature is, of course, the membrane on its sides that enables it to glide. These membranes are not wings per se. They are supported by bony rays of dermal origin and involve neither ribs nor limbs. When it spreads and stretches its upper limbs, this action stretches the membranes that enable it to glide. According to paleobiologists, its ancestor must have jumped from branch to branch to move from one tree to another. In this way, he avoided taking too many risks on the ground. Evolution would later give him the ability to glide between two trees, gaining in agility. It doesn't fly yet, Coelurosaurus only glides, but it opens up new perspectives, those of the conquest of the sky by reptiles. Despite all these radiations and great biological revolutions, the Permian also marks the end of an era, that of the Paleozoic. A mass extinction is underway and will once again upset the order of living things. Many Permian animals will not reach the next era, the Triassic. A new system, the Mesozoic, begins. Before entering this new period, the world will face a major fracture, one that will leave its mark on the Permian and Triassic eras. Pangea will break up again. Yes, it's not the first time that the Earth's internal activity has beaten time and played its most beautiful symphony, the waltz of the continents. This waltz continues, but this time it's no longer about unification, but separation. The North Atlantic Ocean will open up, causing the continent to break up. This fracturing of the land will lead to major changes to which not all species will be able to adapt. Several factors combined over several million years will lead to the disappearance of almost 90% of marine and terrestrial species. It's a chain reaction that will undermine the very cycle of life itself. The food chain breaks down, a domino effect that will impact every member of this chain, notably 75% of terrestrial vertebrates, but also the majority of marine animals. Water contains methane hydrate, also known as clathrate. Until now, it has been in solid form. Pressure and low temperatures have enabled methane hydrate to maintain this state. As the temperature rises, it becomes gaseous, releasing the methane. If carbon dioxide is a powerful greenhouse gas, methane is even more dangerous, because it is even more powerful than CO2. It heats up the water. The ocean gains 10 degrees. This is enormous. The marine breeding ground is transformed into a veritable aquatic hell. The heat also has an effect on the oxygen present in the water. If a few animals had managed to cope with the new temperatures, the lack of oxygen, the anoxia, finishes them off, leaving them no chance of survival. 96% of marine species disappear. Some seabeds are weakened by plate movements, but also by areas where the Earth's mantle has not regenerated sufficiently. The ground is so fragile in places that it causes the ocean floor to collapse in on itself. The sea level will then drop by 250 meters or 820 feet. Where the sea once fringed the coastline, kilometers of bare land appear. The animals benefiting from the waters of the continental shelf will disappear, along with their ecosystem and their best way of life. There is also the hypothesis that meteorites hit the Earth in the Permian, one in Australia and the other in Antarctica, with terrible consequences. But it's difficult, almost 250 million years later, to be certain of the latter. While living organisms were mostly simple, made up of individual cells, or sometimes grouped together in colonies, a veritable zoological Big Bang occurred during the Cambrian, between 541 and 530 million years ago. This explosion of organisms led to the sudden appearance of most of today's major phyla of multicellular animals. This event initiated a gigantic diversification of animal, plant, and bacterial classes and species. The Earth consists of a single continent, Rodinia, surrounded by an immense ocean called Morovia. But this supercontinent is beginning to break up, and new pieces of continent are appearing. The largest of these will occupy most of the southern hemisphere, Proto-Gondwana. To the north, the other pieces form a large archipelago with Laurentia, Baltica, and Siberia. The planetary climate is warm, around 25 degrees Celsius or 77 degrees Fahrenheit. In the center of Laurentia, Baltica, and Siberia, an ocean called Iapetus forms. In its shallows, animal life explodes. Most were soft-bodied animals such as worms and sponges, but there were also the first arthropods, distant ancestors of today's insects and crustaceans, ancestors of future cephalopods, as well as animals that had no descendants. They all evolved on the ocean floor. This new fauna now possesses internal and external organs such as eyes, legs, digestive system, antennae, mouth apparatus. A food chain now exists. Microorganisms such as trilobites form the bottom of this chain, and at the very top is a super predator, Anomalocaris. Anomalocaris is a genus of arthropods and the first known predator in the history of life. Its distinctive feature is its large, complete visual organs, compound eyes. 2 to 3 centimeters or 1 inch long, they have 16,000 facets. Its eyesight, though primitive, enables it to distinguish potential prey, which is already a formidable evolutionary asset. Its long, narrow body measures less than a meter. It is covered by a mineral-free exoskeleton and possesses a toothy mouth, flexible legs, and a long V-shaped tail, which must have enabled it to propel itself swiftly through the water. Its 28 lateral fins are arranged in pairs. Alongside him are many other animals, each more original than the last. Less spectacular in terms of size, Hallucinogea sparsa has a long, slender, worm-like body, measuring up to 3 centimeters or 1 inch. A predatory ancestor of cephalopods, Nectocaris pteryx, is described as having a head with 2 eyes, 1 or 2 pairs of appendages, and an oval carapace at the rear. But the specimen that really catches our attention is the famous pangea, a species of worm or eel just a few centimeters long. Its protodorsal spine suggests that it was the first animal of the chordate phylum, or even the precursor of the vertebrate branch. Today, however, there is no unanimity on this last point. This animal has a chord, a primitive spinal column running the length of the animal. It is from this chord that the muscles and organs that make up the animal are assembled. In the space of a few million years, all the pieces needed for the great adventure of the evolution of the animal world were in place. This Cambrian explosion was halted at the end of the period by an extinction caused by glaciation and a sudden change in temperature. Nearly 40% of species became extinct. Although ecological niches were left vacant, they were rapidly taken over by surviving species during the Ordovician period, leading to evolutionary radiation. Glaciation lowered sea levels and increased the number of shoals. The surviving species multiply, and although the ocean is calmer, it soon becomes populated by animals of all kinds. The families and genera of marine organisms multiply. This is the event of the Great Ordovician biodiversification. The rapid diversification of plankton favored the development of marine fauna living on the ocean floor. Crinoids, bivalve brachiopods, and trilobites multiply, but new arrivals also develop. Corals colonize the oceans and build coral reefs. They enrich the marine fauna. Land seems lifeless, and yet, on closer inspection, a few lichens are beginning to set their sights on it. A lichen is a fungus associated with an algae that grows where no plant can live. The fungus and the algae live in a symbiotic relationship, rather like coral. The fungus protects the algae. It protects it with water and mineral salts. The algae produces sugars and other products that it shares with the fungus. They can be found on rocks by the sea or on cooled lava. At first sight, nothing very extraordinary, and yet, when they rot, lichens leave debris that forms a soil rich enough for moss and other plants to grow. The ground is slowly prepared. The tiny advance of life on Earth is about to be halted by a mass extinction. Glaciation sets in, wiping out many species and slowing down the momentum that some life forms were beginning to build up out of the water. The ocean, once teeming with life, is now calm. Many families, genera, and species of animals and plants have disappeared. There's a kind of void, but nature hastened to fill it, and the surviving species underwent an evolutionary radiation. The continents continue to move. Siberia, Baltica, and Laurentia, which had formed a mega-archipelago, moved closer to Proto-Gondwana. Over time, they formed the supercontinent Laurasia. The collision of these lands gave rise to a gigantic mountain range, the Caledonian Chain. Then the ice began to melt. Ocean levels rise, and certain species develop considerably. With the ice cap gone, the climate becomes mild again, with temperatures around 20 degrees Celsius or 68 degrees Fahrenheit. What about the mainland? We find the lichens that were already starting to appear before the extinction. The first mosses are now appearing, and the flora will begin to establish itself out of the water. While life abounded in the depths of the sea, the landscape on land at the time consisted of bare, windswept rock as far as the eye could see. Then, as they pass, high and low tides leave sediments at the water's edge. This sediment is the first holdfast for plant life. The oldest known trace of primitive plants is that of Cooksonia. High and low tides and the consequent alternation between the passage of water and the drying out of the water allow the flora to gradually become accustomed to life outside the water. Cooksonia plants grow to less than 10 centimeters in height, and have a relatively simple structure. They have no leaves or flowers, only a stem that divides into several branches. Each branch ends in a rounded sporangium containing the spores. A priori, it has no roots and is attached to the ground by radicals, fine superficially concentrated roots. Cooksonia plants release their spores into the air to reproduce. Cooksonia and the other primitive plants are confronted with the phenomenon of gravity, a physics that is new to them. They evolve by developing a hydraulic internal skeleton and a protective layer of wax. They are better able to support their own weight and cope with drought. The cuticle prevents the plant's water from evaporating, but it also forms a layer impermeable to carbon dioxide and oxygen. Over time, plants will develop tiny openings called stomata, enabling them to breathe through the air. Some have cells to distribute water and nutrients to higher organs, but at the time of the first plants, the soil was poor in nutrients. So plants develop rhizomes, like roots, to draw the food they need from the ground. Even though the soil was still almost non-existent, plants themselves helped to build it by breaking down the rocks on which they ventured. A little later, they develop outgrowths to increase the surface area exposed to solar energy. All these primitive plants must also find a way to reproduce. They cannot, like marine plants, discharge their gametes into the water. They must now find an aerial means of reproduction. This is done via spores. These are spread by the wind, and as they germinate, produce male or female organs, which in turn release their sex cells into the moist soil. The higher the plant, the more efficient the spore dispersal. This is why some plants try to grow taller, which also gives them the advantage of capturing even more of the sun's energy than their ground-level counterparts. Now, they need to give their stems more strength to support their weight and height. To this end, they add a woody support tissue. They can grow taller and taller and develop an ever-increasing diameter. Plants reach ever greater heights, with trunks measuring at least 10 centimeters or 4 inches in diameter. This may seem ridiculous compared with our present-day forests, but this primitive vegetation has nonetheless allowed other visitors to venture onto dry land. A few critters docked and ventured out onto the newly forming vegetation. But who are these curious little creatures? Probably earthworms, as life teems in the mud, or even mites. But the first real fossilized traces discovered are those of an extinct species of arthropod. The ebb and flow of the sea washes all kinds of organic waste and corpses onto the shore. This highly attractive food is a great incentive for carnivorous organisms to leave the waters. What's more, the desert is slowly becoming clothed in mosses, lichens, and other primitive plants, a tempting exploration ground for certain organisms. Curious, tiny, and discreet inhabitants shelter between grains of earth and roots beneath a layer of decomposing vegetation on which they feed. Primitive insects swarm in the midst of this growing vegetation. At first, the largest are no bigger than a few millimeters. They look like springtails, the pan-crustaceous jumping arthropods that populate our land today. Some are carnivores, feeding on the corpses of other animals, and others feed on plants, but none seem to attack them until they have rotted. Living plants certainly contain toxins they couldn't survive. It is these little beasts, so insignificant at first sight, that will eventually populate the whole Earth. Today, there are over 1.3 million known species of insect. They occupy almost every ecological niche. But this dominance didn't happen overnight. Although insects diversified very early on, in the Carboniferous period, they were initially present in small numbers. Campacaris obanensis belongs to the myriapod genus, closely related to millipedes. Its fossil was discovered in Scotland. Its legs have not been preserved. The animal measures approximately 2.5 centimeters or 1 inch in length. Its body is segmented. It evolved in an environment close to bodies of water, and certainly fed on decomposing plants. Although not their ancestor, they bear a striking resemblance to today's millipedes. Its body is covered by a carapace that is synthesized by its glands. Cooksonia fossils have been found in the same region. Strudiella devonica is a species of fossil arthropod. It is one of the oldest terrestrial insects found and the only complete fossil insect. This small insect, measuring around 8 millimeters in length, appears to be a close relative of the early grasshoppers. It has a relatively large head, with robust mandibles and long antennae. This specimen is wingless, which prevents scientists from knowing whether it is a larva or an adult. Strudiella devonica has all the morphological features and organs required for terrestrial life. Its unspecialized mouth parts indicate that it had an omnivorous diet. The fossil shows 6 body segments, 6 legs, and 2 antennae. The genitalia have not been preserved. The first arthropods to set foot on Earth were faced with a number of difficulties. They needed a structure strong enough to support them, as well as arrangements to protect them from ultraviolet light and desiccation. They also needed to find food. Surprisingly, most arthropods are carnivores, and few are herbivores. And to satisfy carnivores, prey must be available. Dry land is still relatively devoid of life, and the first arthropods may have had to make do with fungi, molds, or rotting leaves. The first plants were therefore not a source of food, but they helped to shape the first ecosystems that gradually spread across the land. At the same time, as life was gaining ground on land, it was demonstrating real innovation in marine organisms. The Devonian is reputed to be the period known as the Age of Fish. Numerous gigantic armored fish, known as placoderms, crisscrossed the oceans, including Bothriolepis, Silurians, and Dunkleosteus. While life on land is represented only by small organisms, life in the oceans is luxuriant. Its evolution was not unrelated to the emergence from the sea. Placoderms are the vertebrates that dominate the marine world. They are highly diversified, frequenting both reefs and the open sea. They feed on algae, mollusks, and other fish. They owe their name placoderm to the plates that cover their skin. Their head and thorax are covered by a thick bony armor that protects vital organs such as the eyes, brain, and heart from predators. Their enormous jaws made of bone with sharp edges, are like pincers that can cut their prey in two. Although placoderms were the first to be equipped with jaws, they still lack teeth. Around 400 million years ago, the group of jawed fish diversified into two major groups. On the one hand, cartilaginous fish, from which sharks, rays, and other chimeras evolved, and on the other, bony fish. Subsequently, bony fish are divided into two groups. Bony fish, known as actinopterygians, have fins with keratin rays, the protein that makes up hair, nails, and horns. The rays are shaped like arms that support the fin fan. Alongside them, bony fish with fleshy or lobed fins are called sarcopterygians. Their fins are characterized by bones that form an articulated internal skeleton. The ventral fin bones are identical in number and positioned to those of the tetrapod extremities. This new form of fin is now articulated around a single element, the other rays having disappeared. It's not yet a leg, but it sure looks like one. The fin ends in a soft veil of scales. Panderichthys is an extinct genus of sarcopterygians that is considered an anatomically transitional form in the evolution from fish to tetrapod. The internal skeleton of the pectoral and pelvic fins is reminiscent of Chiridian limbs. The best-known species is Panderichthys rhombolepis, which resembles a modern-day catfish. It measures between 90 and 130 centimeters or 50 inches. It has primitive lungs. This species marks an important stage in the adaptation to terrestrial locomotion, since its fleshy pectoral fins are prototypes of tetrapod limbs. This suggests that the transition to tetrapod locomotion, which relies on hind limb propulsion, went through a stage of forelimb support accompanied by body undulation. In fact, the pelvic fins of Panderichthys rhombolepis are primitive and do not allow it to take support. A few million years later, the veil at the end of the fin would evolve into a revolutionary structure in living organisms: fingers. Meanwhile, vegetation continued to grow. Primitive plants have grown so much that they can now be called trees. It took a good 50 million years for the first giant ferns, calamites, and lepidodendrons to reach heights of over 30 meters or 100 feet. At this size, the trunk is reinforced with wood and bark, like that of today's trees. The arborescent form appeared independently in different plant groups. Archaeopteris is like a fern with a coniferous trunk. It thrives in well-drained areas where it can develop its extensive root system. It contributes to the formation of deep soils. It can reach heights of up to 40 meters or 130 feet, and measure up to 1.5 meters or 5 feet in diameter. Its wood is similar to that of conifers, with tracheids and rays. Many scientists have studied the anatomy of its trunk, making it the first modern tree. The wood of Archaeopteris becomes both a conductive tissue and a support element, an ideal combination for seeking light at height. Watieza is a close relative of modern ferns and horsetails. It is one of the components of the oldest forests and is considered the oldest arborescent plant. Its general appearance is reminiscent of modern palm trees or tree ferns. It can reach heights of up to 8 meters. Instead of leaves, Watieza has fronds located in the crown in a vertical position and positioned helically on the trunk. In some plants, fronds play the role of leaves. They are often large and divided into segments. As it grows, Watieza loses its fronds, leaving scars on the surface tissue of the stem. The trunk is rather thin, with grooves in the area around the crown, the scars of the lost fronds. This tall plant remains upright thanks to its inverted conical morphology and the large number of small roots that penetrate perpendicularly into the soil. One representative of the first forests is the Gilboa petrified forest in the United States. This forest was discovered during the excavation of a quarry to build the Gilboa Dam. Hundreds of large fossilized stumps preserved in the rock were identified. It would take several years of study and research to understand this forest a little better. The bases of the trees making up the forest are bowl-shaped, and their diameter can reach up to 2 meters or 7 feet. Thousands of roots surround them. According to estimates, the tree could reach up to 10 meters or 33 feet in height and resemble palm trees or tree ferns. Horizontal woody stems, measuring around 15 centimeters or 6 inches in diameter, were also discovered. These are creeping trunks of a neurophytalian progymnosperm, previously known only for their upright branches. A large specimen of lycopod was also discovered. This herbaceous plant has axes covered with small, narrow, microphyllous leaves. Fertile leaves have 5 segments, like vegetative leaves, and sporangia are inserted on the upper surface of each. This primitive forest already displays extraordinary complexity. The Tiktaalik roseae is the only known species of Tiktaalik, which literally means great fish of the low waters. The Tiktaalik has all the characteristics of a fish. It has scales and gills, but its triangular, flattened head and unusually shaped fins stand out. In fact, in addition to the fine ray-shaped bones that all fish have for paddling, the fins have much stronger internal bones, enabling the animal to wedge itself in shallow water using its limbs for support. This is what most four-legged animals do. This particular fin makes Tiktaalik a transitional species between fishes and Pandarictus, and the future first tetrapods, Acanthostega and Ichthyostega. The fins of Tiktaalik roseae have wrist bones and simple finger-like rays. It has gills but also primitive lungs. This enables it to establish itself in shallow waters and on land. Its scales are diamond-shaped, its teeth are structured in 2 rows, and it has internal and external nostrils. Most fish have bony plates in the gill area, limiting head movement. Tiktaalik roseae has none. This makes it the first known fish to have a neck and pectoral girdle separate from the skull. This gives it greater freedom to hunt prey, whether in the water or on land. The rays of Tiktaalik roseae's pectoral fins, those that evolved to form the front legs of tetrapods, have become more robust, and an asymmetry has developed between the rays on the ventral and dorsal sides. The latter become wider and more numerous than the former. This characteristic has resulted in a fleshy, palm-like part that the animal can use to hold itself up on the ground or in rivers. This suggests the presence of muscles under the fins to support the animal's weight when pulling itself up in the open air. Another visible evolution on the animal is that its eyes and nostrils are positioned above the head. As brackish waters are low in oxygen, it is forced to breathe at the surface. This feature enables it to breathe without taking its head out of the water, like today's crocodiles. Tiktaalik rosea is somewhere between a fish and a tetrapod. Its anatomical characteristics make it an intermediate species. Acanthostega and Ichthyostega were among the first tetrapods, defined as legged fish. They were aquatic animals with gills, but their 8-toed legs enabled them to navigate muddy, cluttered environments out of the water. Acanthostega is considered one of the first terrestrial vertebrates. It has chiridial limbs, articulated locomotor appendages equipped with fingers, an anatomical feature of the first tetrapods. Acanthostega measures around 60 centimeters in length. On each of its hands it has 8 fingers linked by a tendinous sheath. It has no wrists but a shoulder and a forelimb similar to those of a fish. However, the shoulder and forelimb cannot be bent forward or at the elbow, and cannot be used for standing or walking. He therefore had to use them for paddling or clinging to vegetation. Its pelvic girdle becomes a load-bearing structure, enabling it to counteract the force of gravity when it is no longer in the water. Although Acanthostega has lungs, its ribs are too short to support the rib cage outside an aquatic environment. Many scientists agree that Acanthostega would never have lived outside the sea and would have been primarily an aquatic animal. It must have come ashore to find food. Its jaw shows a difference from that of fish, which would have enabled it to feed on food either out of the water or on land. Alongside Acanthostega, Ichthyostega is one of the oldest known four-limbed vertebrates. Ichthyostega is a rather large animal for its time, measuring around 1.5 meters or 5 feet in length. Its eyes are set on top of a flattened skull, and it has large teeth. Its limbs are long, with 7 toes on each end of the hind limbs. The morphology of its shoulder and pelvic girdles is more robust and better adapted to terrestrial life than that of Acanthostega. Its vertebrae are also stronger. How did the fingers get added to the limb tips of these early tetrapods? Were the fingers pre-existing elements, or did they transform and evolve over time? The latest research suggests that the fingers originated from abnormal growth of the fin tips. In the fish embryo, the fin tips form the apical ridge through which the bony rays of the fin are formed. Once all the rays have formed, the ridge peels off, stopping the construction of new bony structures on the fin. The fin then forms a veil and completes its development. The anomaly that led to the development of the fingers would have been that the apical ridge failed to detach. After forming the bones of the arm, forearm, and wrist, it then went on to build the phalanges. With their fingers, Acanthostega and Ichthyostega were able to survey muddy deltas dotted with plants and tree branches. They were able to spread branches and wade through mud, as well as move from one arm of a river to the other. Their life remained essentially aquatic. Their legs are still not very functional, as the absence of joints makes them very rigid. As they have no ankles, they are unable to extend themselves. These protopatties are therefore only good for carrying fish and being used as paddles for travel. Finally, at the end of the Devonian period, 360 million years ago, these legs came in very handy. Terrestrial environments became increasingly vast as ocean levels dropped. Acanthostega and Ichthyostega, along with their tetrapod congeners, are able to reach isolated bodies of water and rivers. They survived this new environment better than others. It was during the Carboniferous period that flexible ankles, wrists, and elbows gave them incredible mobility. They could now leave the water's edge and venture out to conquer the immense forests that had in the meantime colonized dry land. Soon after Acanthostega and Ichthyostega, sea scorpions and spiders reached dry land. But this conquest was halted by another massive glaciation, which led to a mass extinction. 75% of the species that existed in the Devonian disappeared. The Earth emerges from a major mass extinction, resulting in the death of many animals. At this time, the continents Laurasia and Gondwana come together to form a supercontinent called Pangea. It is surrounded by the Panthalassa Ocean. Tectonic plate movements cause sea levels to rise. Many of the plants that remain on dry land are drowned. But nature has not said its last word. Forests continue to colonize. In the midst of tree ferns and giant horsetails, a new tree has appeared, the Lepidodendron. It's one of the giant lycopods, oversized plants that have dominated the Earth for a long time. The Lepidodendron reaches heights of over 50 meters or 165 feet, with a trunk 2 meters or 7 feet in diameter. Its trunk is firmly anchored to the ground by numerous roots. It has no intermediate branches, but is topped by a crown of foliage arranged like a parasol. This is when bark and seed trees appear. Bark trees became the source of coal, which was used in abundance during the Industrial Revolution. Following fires or floods, these trees were buried and formed sediments. Deprived of air and subjected to high pressure, organic elements are transformed into carbon compounds. The emergence of seed plants was a botanical revolution and a new step in the development of life on land. Henceforth, plant reproduction was no longer dependent on water and became aerial. The elements that ensure the plant's dissemination are no longer spores, but seeds. These multicellular seeds are protected by protective envelopes formed by the lignified integuments of the ovule. They contain not only an embryo, but also the reserves such as carbohydrates, lipids, and proteins needed for its development during the early stages. The seed detaches from the plant and ensures the dissemination of the species. Their tiny size and hooks enable them to attach themselves to animals, while their appendages provide a grip on the wind, all characteristics that facilitate dispersal. Seeds also have another fundamental characteristic. Their tissues are dehydrated, allowing them to enter dormancy when conditions are unfavorable for germination. Seeds can therefore be buried in the soil for several years before germinating. Animals continue to develop and adapt to dry land. In the mid-Carboniferous, sea levels drop again. The extension of terrestrial environments isolates bodies of water. But tetrapods now have limbs that can bend, and they adapt very well to this new environment. They also have lungs and are ready to conquer the gigantic Carboniferous forests that offer them a moist, shady environment in which to live. Amphibians were the first tetrapods. They were able to live both in water and on land. They had lungs to breathe air, but they also had to lay their eggs in water. But a new phylum appeared during the Carboniferous period, the reptiles. These were the first tetrapods to be fully adapted to life on land. They had a body covered in scales that protected them from predators and the elements. They also laid their eggs on land. Insects developed rapidly during this period. Insects have diversified very rapidly since they first appeared. By the Carboniferous, they accounted for at least 60% of biodiversity. In just a few million years, this group outnumbered their main terrestrial competitors. Their secret? A very useful feature for survival in this world of predators: wings. The group of wingless insects known as apterygotes represents just 1% of today's insects. Their congeners have largely overtaken them. The appearance of wings is difficult to date and explain. Perhaps they were gills, modified to enable aquatic organisms to fly short distances? This is just one hypothesis among many. What is certain is that thanks to their gravity-defying wings, insects were the sole masters of the air for several million years. Although flora, with the appearance of seed plants, gradually freed themselves from the aquatic environment, animals remain dependent on water for their food, and above all for their reproduction. A major innovation occurred around 310 million years ago with the appearance of the amnion. This is the envelope that allows the amniote embryo to develop in a liquid environment. Its innovative feature is that it can be laid out of the water. The embryo is thus protected from drying out and shocks. At the same time, some animals are equipped with a dry, more or less watertight skin to slow down water loss through the skin. Several groups of vertebrates, such as reptiles, birds, and mammals, were equipped with an amniotic sac. These groups of animals can now move away from the aquatic environment, as can amphibians, which remain water-dependent. The amniotic egg combined with the waterproof skin of the first reptiles enabled them to venture into continental areas previously inaccessible to them. They diversified, giving rise to numerous groups such as crocodilians, turtles, lizards, snakes, birds, and mammals. The appearance of the first flowering plants in the history of life on Earth is very difficult to date, as they are difficult to fossilize. Nonetheless, they play an essential role for the plants that bear them, attracting insects for pollen dissemination. Exceptional paleontological deposits have led to the discovery of two extremely well-preserved flowers. The first, Archifructus, resembles today's water lilies, but its anatomical organization remains unclear. The second, Montsechia, has long, narrow stems. Numerous fruits are found throughout the plant. These primitive flowering plants have little in common with the more classic flowers we know today. Since they first appeared, flowering plants have continued to diversify. Today, there are over 270,000 species in all environments, whether on land or in water. They range in size and shape from orchids to oak trees. By comparison, the fern group comprises 10,000 species, and the pine and associated species group around 750. And yet, these two groups are much older than flowering plants. The latter are therefore a true evolutionary success story. This is perhaps due to the way flowering plants reproduce, enabling them to rapidly colonize all types of environment. Fauna and flora are now well established on land. Although they will suffer disasters over the years, leading to more or less profound modifications, nothing will totally wipe them out. What are the effects of this outflow of water on the Earth? The colonization and establishment of life on the continents caused major changes to planet Earth over hundreds of millions of years. Land plants were the first to colonize the Earth outside water. Their roots contribute to soil formation. They break up rocks, facilitating the concentration of organic matter. Soils are essential for plant growth and have important geological implications, particularly with regard to erosion and land stability. Plant roots help prevent soil erosion by stabilizing the soil. Forests and meadows help to slow the flow of water. Particles transported by watercourses can thus be sedimented. This contributes to the formation of sedimentary rock. Certain living organisms, such as lichens and bacteria, promote the chemical alteration of rocks by releasing organic acids that can dissolve minerals. As a result, rocks disintegrate and soils are formed. Living organisms are mainly made up of carbon, hydrogen, nitrogen, and oxygen. When they die, organic matter is decomposed by microbial activity. If this happens in an aerobic environment, all the carbon is converted into carbon dioxide, and mineralization is complete. If, on the other hand, this takes place in an anaerobic environment, mineralization stops as soon as all the oxygen initially present has been consumed. Most of the biomass undergoes total mineralization. The remaining 1%, which sediments, is the source of oil, natural gas, and coal. In this way, the accumulation of plant debris in rapidly sedimenting areas during the Carboniferous period created large reserves of carbon that are used today in the form of coal. The weathering of limestone can then be accelerated, leading to the formation of caves. Caves are underground voids of varying sizes. Microbial activity in the soil contributes to the production of carbon The appearance of life and its emergence from the waters involve complex interactions between chemical elements, geological processes, and living organisms. This has had a major impact on the establishment and evolution of biogeochemical cycles. These cycles are essential to the regulation of ecosystems and the survival of life on Earth. Biogeochemical cycles are the processes by which elements or chemical compounds are transported or transformed between the major terrestrial reservoirs, the geosphere, atmosphere, and hydrosphere, in which the biosphere is found. Through a series of processes, elements move from one medium to another, returning to their original environment in an infinite recycling loop. Living organisms need a constant and regular supply of essential elements such as carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur to ensure their survival. Elements circulate continuously from one environment to another, from one form to another. They are found in soil, atmosphere, water, and living tissue. This continuous circulation is known as the biogeochemical cycle. These cycles combine biological, chemical, and geological processes. Biological processes are defined as respiration or digestion. Chemical processes are combustion or synthesis reactions. The latter two take place over relatively short periods of time. Geological processes, on the other hand, occur over much longer timescales and are defined by erosion or sedimentation. The establishment of biogeochemical cycles on Earth is a complex process that has developed over billions of years. These cycles have evolved in response to interactions between chemical elements, geological processes, living organisms, and terrestrial, aquatic, and atmospheric environments. The colonization of land by plants and animals has had a major impact on biogeochemical cycles. Many mechanisms are involved, but let's take a look at the four most important cycles. The carbon cycle. The significant greening of continents apart from the polar and desert regions is a major change in Earth's history. This conquest of land by plants is not without consequences, as terrestrial plants are important players in the carbon cycle. Plants use carbon dioxide from the atmosphere to form their own living matter through the process of photosynthesis. In addition, their root cells breathe and release some CO2 into the soil. This creates acidic solutions in their vicinity and encourages the weathering of rocks, which in the long term traps carbon dioxide in the atmosphere. The development of terrestrial vegetation also deepens soils by a few centimeters to 1 meter on average. The increase in continental vegetation has reduced the greenhouse effect and cooled the climate. In the long term, dead organic matter accumulates on the soil in the form of plant biomass, helping to form soils rich in organic carbon. This is then buried and transformed into fossil fuels, such as coal, oil, or natural gas. This transformation requires soil carbon sequestration. The nitrogen cycle. The massive arrival of plants on land has also had an impact on the nitrogen cycle. Nitrogen is the most abundant gas in the atmosphere. Today's air contains 78% of it. This element is essential to the functioning of living organisms, since it is used to make proteins and produce the nitrogenous bases present in DNA. However, it cannot be directly assimilated in its initial form by most living organisms. We must therefore rely on bacteria to transform atmospheric nitrogen into a form that can be assimilated by other organisms. Through its biogeochemical cycle, nitrogen passes from one form to another. Bacteria, such as cyanobacteria and rhizobium, which live in symbiosis with plants, capture atmospheric nitrogen and transform it into ammonia, which is then used directly by plants and animals and reacts with hydrogen to form ammonium. Oxidation reactions then come into play, with some bacteria oxidizing ammonium to form nitrite, while others oxidize nitrite to form nitrate. Transported in soil or water, nitrate and ammonium are captured by plants through their roots. Nitrogen now fixed on plants is available to herbivorous animals and is passed on in the terrestrial food chain. Carnivorous animals ingest nitrogen by feeding on herbivorous animals. Nitrogen is then present in plant and animal waste. Certain fungi and bacteria decompose it and produce ammonia, which dissolves to form ammonium. Certain bacteria, known as denitrifying bacteria, convert nitrate into dinitrogen, returning it directly to the atmosphere. The phosphorus cycle. The phosphorus cycle was greatly disrupted when living organisms colonized dry land. Phosphorus is essential to life. It is a basic material in DNA, bones, shells, and teeth. The phosphorus cycle is sedimentary, meaning that it does not involve atmospheric processes, as it has virtually no gaseous components. The main source of phosphorus is the erosion of sedimentary rocks. Under the action of rain and wind, small quantities of phosphorus escape from the rocks. It is released in the form of dissolved phosphate and enters the food chain, starting with plants. The latter directly assimilate this essential element for their growth. Herbivores then feed on the plants, which are then eaten by carnivores. Phosphates are returned to the soil via animal feces and urine, as well as through the degradation of animals and plants once they have died. However, over time, some phosphorus has been leached from the soil and transported to the oceans, where it has become trapped in marine sediments. Human activities have resulted in additional phosphorus inputs. The mining of these phosphates for fertilizer production has altered the phosphorus cycle, leading to environmental disturbances such as eutrophication of watercourses. Water quality declines, affecting the ecosystem and the aquatic environment. The water cycle. The proliferation of terrestrial vegetation has influenced the water cycle by promoting evapotranspiration. This is the process by which terrestrial liquid water is returned to the atmosphere in gaseous form. This water comes from the sublimation of snow, the evaporation of free water, or water buried in the ground, and also from plant transpiration. How do plants, and trees in particular, transpire? You've probably noticed how nice and cool it is in the heart of a forest when it's hot. A tree actively cools the surrounding air, like a parasol, which simply reflects the sun's rays. A tree combines both actions. To grow, plants need nutrients and CO2, which they absorb and store through photosynthesis. Sap circulates through the tree, transferring mineral salts to the leaves for photosynthesis. Transpiration is the driving force, transporting water and nutrients to the leaves. Sap flows are regulated according to plant needs and climatic conditions. Transpiration takes place in the leaves, at the stomata, and in the lenticels on the bark of trunks and branches. Depending on environmental conditions, stomata may close to avoid excessive water loss. Nevertheless, the quantity of water released by a tree during evapotranspiration is very significant. For an oak tree, this amounts to 1,000 liters of water per day. Evapotranspiration is an essential component of the water cycle. Although it represents only a small part of the overall water cycle, it is fundamental. It transfers water from the soil and vegetation to the atmosphere. At present, urbanization, deforestation, and other practices that make soil impermeable reduce evapotranspiration and alter the water cycle by altering the infiltration of water into the soil. The consequences are flooding, reduced groundwater reserves, and changes in precipitation patterns. Life could not emerge from the water until it had a shield to protect it from the sun's ultraviolet rays. This is the ozone layer. Oxygen is present in 3 forms in the atmosphere. There are oxygen atoms, O, oxygen molecules, O2, which represent the oxygen we breathe, and ozone, O3. Ozone is a molecule made up of 3 oxygen atoms. It is a colorless gas, but has a very strong odor. Ozone is formed in a section of the atmosphere known as the stratosphere. In this region, high-energy solar photons can strike oxygen molecules and separate the 2 oxygen atoms. When a free oxygen atom comes into contact with an oxygen molecule O2, an ozone molecule is formed. Ozone is then naturally degraded in the stratosphere through a series of chemical transformations under the effect of the sun's ultraviolet rays. In an unpolluted atmosphere, ozone production and destruction exactly balance each other out. Its total concentration then remains relatively constant. Ozone naturally accumulates in the stratosphere, but is only present in abundance when there is sufficient oxygen. Scientific research estimates that the first threshold of the ozone layer was formed around 600 million years ago. This first threshold allowed an explosion of life during the Ediacaran period and later during the Cambrian. It was then present at 90% of today's ozone levels. The second threshold was reached around 400 million years ago, coinciding with the emergence of the first animals from the waters. The molecules making up living matter are photosensitive, which means they can be destroyed by certain types of radiation, such as ultraviolet light. Ozone absorbs a large proportion of ultraviolet rays, protecting living organisms from their harmful effects. When the Earth was first formed, it was covered in volcanoes, and the atmosphere consisted mainly of carbon dioxide and methane. There was no oxygen or ozone. In those days, without an ozone layer, the sun's ultraviolet rays reduced all organisms to aquatic and simple single-cell life. Around 600 million years ago, the accumulation of oxygen by cyanobacteria and other oxygen-releasing organisms enabled the atmosphere to reach a first threshold of oxygen, and thus ozone. When the first terrestrial arthropods emerged from the waters around 400 million years ago, the second threshold, equivalent to the present one, was reached. The oxygen produced by living organisms such as plants and the first forests slowly reached the upper layers of the atmosphere, where it was degraded by ultraviolet radiation, giving rise to ozone. Ozone supports the development of life when it is present in the stratosphere, above 15 kilometers or 9 miles in altitude. Ozone concentrations peak at around 25 kilometers or 15 miles above sea level. It is precisely this ozone-rich region of the atmosphere that is known as the ozone layer. Moreover, the amount of ozone present at any given point on the Earth's surface varies according to day, season, and latitude. The emergence of life forms from the water represents a quantum leap in the evolution of life on Earth. If this has been possible, it's because life on land has many advantages over life in the water, although this largely depends on the needs and preferences of the organisms in question. Terrestrial organisms have direct access to atmospheric air, which is far more abundant in oxygen than that dissolved in the aquatic environment. This facilitates air breathing. What's more, air is less dense than water. Land-dwelling organisms don't have to fight against buoyancy, allowing for a greater diversity of body shapes and structures. On land, solar energy is harnessed to a greater extent, and body temperature is more easily regulated. Its direct use for photosynthesis is an advantage over aquatic organisms, which receive limited energy from the depths. The visual pathway is undeniably one of the wonders of nature. The ability to see near and far, in black and white and in color, while embracing a wide field of vision, certainly represents an evolutionary advantage of remarkable, almost miraculous magnitude. To understand the history of vision, we need to go back in time to explore the theories of eye evolution. Step by step, we understand how this organ is developed over time. Scientists have been divided into two camps. On the one hand, there are those who believe that eyes had a polyphylogenetic origin, meaning that the eye would have evolved independently several times in different species. On the other, those who believed in a monophylogenetic theory, asserting that all eyes derive from a common ancestor, and that each eye type had evolved separately according to the specific needs of the organism. According to recent studies, the trend favors the second theory, i.e., the monophylogenetic origin of eyes. Genetic research has revealed that the PAX6 gene, responsible for light detection, is common to all sighted species. The presence of all three eye types in mollusks intrinsically suggests a monophylogenetic origin of this visual organ. Exactly how long it took for these developments from a single ancestor species to occur remains unknown. Biologists have identified 4 key stages in the evolution of the eye: emergence of efficient photopigments, acquisition of directionality, formation of the photoreceptor membrane fold, and development of optical focusing ability. "600 million years ago, the first organisms developed the most primitive visual organ, consisting of a single photoreceptor, composed of two distinct chemical substances, a light-sensitive receptor and a pigment. Its unique function was to detect the presence or absence of light, acting as a kind of electrical switch, with only two possible states, on or off. The use of this structure was relatively simple, as it was not used for the perception of movement or localization like our modern eyes. Instead, its role was dedicated to a process called photoperiodicity, which was equivalent to the modern circadian rhythm. In the presence of light, certain reactions occurred, while in its absence they were inhibited. Let's meet a specimen equipped with this very first visual system. This marine animal is very primitive. The hagfish resembles an eel and has no jaws. The hagfish's eyes are very simple and resemble the pineal organs found in non-mammalian vertebrates, especially that they are positioned on either side of its head. Curiously, despite these eyes, the hagfish appears almost blind and reacts very little to light, even when its eyes are removed. It would seem that hagfish eyes aren't really made for vision. In fact, they seem to play a role similar to that of a circadian organ, i.e., they could help it regulate its biological rhythms, like our internal clock. A remarkable example of the evolution of the vision system can be seen in the developmental changes observed in the eyes of lampreys. These changes support the hypothesis that the lamprey's ancestors succeeded those of the hagfish in the course of evolution. Indeed, the Amocoetes, or lamprey larvae, have eye characteristics similar to those of the hagfish. In the larval stage, its eyes are small, sunken under the skin, and have a relatively undifferentiated retina, giving it very limited or non-existent vision. Over time, however, a slow process of neuronal differentiation takes place in a specific order. Ganglion cells, followed by amacrine and horizontal cells, then photoreceptors, and finally bipolar cells are formed. This sequence of differentiation is similar to that observed in jawed vertebrates for the development of their retina. When metamorphosis occurs over hundreds of thousands of years, the lamprey's eye undergoes significant transformations. It enlarges considerably, its retina differentiates completely, the lens develops, the cornea divides into scleral and dermal layers, allowing the eye to move relative to the epidermis. As in jawed fish, extraocular muscles develop, and finally, the eye bursts through the surface to form a visual organ similar to that of vertebrates. The process of eye development in lampreys suggests that these animals have inherited their visual apparatus from a common ancestor they share with hagfish. The existence of a hagfish-like larval eye, which transformed into a vertebrate-like eye in adulthood, supports the idea that lampreys evolved from a common ancestor with hagfish. The next step in the evolution of the eye was the fusion of these photoreceptors to form an eye spot. This advance enabled the organism to detect the intensity of light. In simple terms, if several photoreceptors were activated in an eye spot, this meant that there was strong luminosity and vice versa. This adaptation improved the perception of surrounding brightness. Once again, scientists have discovered clues in embryonic development. Early in the process, a neural structure develops and gives rise to the eye, forming two sacs, or vesicles, on either side. Later, each vesicle coils to form a C-shaped retina, which lines the inside of the eye. It's very likely that evolution followed a similar pattern. At a later stage in embryological development, as the retina folds inwards, the lens forms, leading to a thickening of the outer surface of the embryo, known as the ectoderm. The ectoderm swells into the curved space created by the C-shaped retina, gradually detaching from the rest of the ectoderm to become an independent floating element. It is likely that a similar sequence of changes took place in the course of evolution. The exact date of this change remains unknown, but in 1994, researchers at Lund University in Sweden demonstrated that the optical components of the eye could evolve relatively rapidly, within the space of 1 million years. If this is the case, it is conceivable that the image-forming eye emerged from the non-visual proto-eye in a short period of time on the geological scale. In parallel with the emergence of the eye spot, organisms also developed locomotion mechanisms such as flagella and cilia. These developments led to the need to localize the light source. To fulfill this role, several eye patches fused together to form an eye patch, like a patchwork quilt. These eye patches enabled the organism to roughly locate the light source and orient itself in its direction. By grouping several eye patches together in this way, organisms were able to improve their ability to perceive and move in relation to ambient light. In order to improve localization, the eye patch had to undergo progressive expansion. However, space was an evolutionary constraint. In response to this challenge, nature came up with a solution: the eye cup, also known as the optical cup. The development of the eye cup in vertebrates proceeds as follows. The eyes take shape from evaginations of the developing diencephalon. Depressions known as optic grooves form on either side of the rostral neural plate. As the neural plate folds upwards and inwards to form the neural tube, these growing regions project outwards and are called optic vesicles. When the expanding optic vesicle comes into contact with the surface ectoderm in a specific region called the lens placode, this induces changes in both tissues. The optic vesicle then invaginates to form the optic cup, while the ectoderm undergoes differentiation to form the lens hollow, which later transforms into the lens itself. It is sometimes suggested that the invagination of the optic cup results from the growth of the lens, but observations show that this cannot be the case. In fact, a normal ocular cup and retina can develop in the absence of a lens, indicating that these developmental processes are independent and follow distinct pathways. This adaptation enabled the organism to precisely determine the angle of the incident light beam according to the photoreceptor activated. Thus, the optic cup played an important role in improving the ability to localize the light source, despite the spatial constraints faced by the organism. By this stage, the primitive eye was fully formed, and all the species that possessed this organ entered into fierce competition for available resources. This competition gave rise to a major evolutionary event, known as the Cambrian explosion, which occurred around 540 million years ago. Scientists argue that the evolution of the eye accelerated rapidly during this period, as precise detection of prey and light gave organisms a survival advantage. So the better the eye, the better the organism's ability to survive. This selective competition for improved visual systems played a key role in the rise in diversity and complexity of animal species during the Cambrian explosion. After the Cambrian explosion, the next period saw the evolution of two essential eye structures: the iris, followed by the lens. The first step was to isolate the eye from the outside environment, which was achieved by the development of a transparent epithelial layer, now known as the cornea. Once the light detection system had been separated, the next evolutionary advantage was the ability to focus. Increased focusing power dramatically improved object localization. The first structure to develop this enhanced focusing power was the iris. Thanks to this adaptation, all incoming light was channeled into a single beam, according to the direction of the light rays, greatly improving the accuracy of light source localization. The next major development was the crystalline lens. This lens allowed light rays traveling long distances to focus directly on the retina rather than behind it. This enabled the organism not only to locate the light source precisely, but also to perceive the shape, depth, and other properties of surrounding objects in greater detail. The lens thus opened up new sensory perspectives for organisms with advanced visual systems. With the advent of the lens to capture light and focus images, the eye acquired a greatly enhanced capacity to gather information. This improvement would have generated selective pressures favoring the emergence of more efficient signal processing in the retina, going beyond the simple connections between photoreceptors and output neurons. To meet this requirement, evolution has modified the cell maturation process so that certain developing cells, instead of becoming ciliary photoreceptors, become retinal bipolar cells, which intercalate between the photoreceptor layer and the output neuron layer. This is why retinal bipolar cells bear a strong resemblance to rod and cone cells, although they lack rhodopsin and are not sensitive to light. Instead, they react to the chemical or neurotransmitter released by the photoreceptors, enabling them to play an essential role in processing visual signals before passing them on to the output neurons. Camera-type eyes offer a wide field of vision, usually around 180 degrees. In practice, however, our brains are limited in their ability to process all the visual information available at any one time, due to the limited number of nerve fibers connecting our eyes to our brains. As a result, we can only sample a fraction of the visual data available. It is likely that the first camera-like eyes faced an even more severe limitation, with probably an even smaller number of nerve fibers. This constraint would have exerted considerable selective pressure in favor of the evolution of muscles to move the eyes. The presence of such muscles must have existed around 500 million years ago, as the arrangement of these muscles in lampreys, whose lineage goes back that far, is almost identical to that of jawed vertebrates, including us humans. Despite the ingenious features of vertebrate eye evolution, a number of rather counterproductive traits can be identified. For example, the retina is positioned upside down, which means that light has to pass through the entire thickness of the retina, including nerve fibers and intermediate cell bodies that scatter light and impair image quality before reaching the light-sensitive photoreceptors. In addition, blood vessels cover the inner surface of the retina, casting unwanted shadows on the photoreceptor layer. Another tricky aspect is the presence of a blind spot, where nerve fibers that cross the surface of the retina come together before tunneling through it to emerge as the optic nerve at the back of the eye. This list of inelegant features could be extended to include other examples. These imperfections are by no means inevitable features of a camera eye, as octopuses and squids have evolved independently of camera eyes which do not have these defects. Examining the vertebrate eye in an evolutionary context highlights these seemingly absurd shortcomings as the consequences of an ancient sequence of evolutionary steps, each of which brought advantages to our vertebrate ancestors, long before they developed advanced vision. The eye has evolved in many different ways over time, giving rise to a variety of features in different species. Jellyfish, for example, have eyes, and although they have no brain, these eyes transmit information directly to the muscles for rapid reaction. Nautiluses have an aperture without a crystalline lens, adopting a different focusing mechanism. Flies, on the other hand, have many eyes, giving them a wider field of vision. The diversity of ocular adaptations continues with predators, which have forward-facing eyes for better depth perception, while prey have lateral eyes, offering a wide field of vision for detecting potential dangers. In the case of humans, they have developed the ability to artificially correct their vision using devices such as spectacles, enabling them to compensate for certain visual imperfections. Ultimately, the list of ocular adaptations is truly infinite, with each species evolving uniquely to suit its environment and specific needs. All the auditory organs of today's terrestrial vertebrates, whether reptiles, birds, or mammals, have evolved in completely distinct ways. Ciliated cells, the sensory cells at the base of the auditory organs of all vertebrates, emerged very early in the evolutionary history of animals. They play a significant role in today's non-vertebrate animals, such as ascidians, and can be traced back to the first animals with well-developed tissues, the cnidarians, like sea anemones. The auditory part of the ear evolved from a vestibular organ whose evolutionary origin, in particular the sensory cells themselves, resembles that of the lateral line, a body surface organ used by fish to detect the movement of fluids in water. The lateral line has sensory hair cells similar to those in the cochlea, whose responses are strongly influenced by viscous damping and are linked by a gel-like structure. The damping problem was solved early on by evolving the active mechanisms of the sensory cells and linking them via a gel-like structure to increase both their sensitivity and frequency selectivity. These two features have survived 500 million years of evolution and are common to vertebrate inner ears. During the Carboniferous period, from -359 to -299 million years ago, tetrapod ancestors successfully migrated to terrestrial life, paving the way for further adaptations of existing sensory systems, including hearing, from aquatic to terrestrial life. Thus, over hundreds of millions of years, progressive adjustments were made to these organisms so that they could adapt to higher frequency stimuli, first in an aquatic environment and then in an aerial one. It is assumed that the ancestors of amniote vertebrates had a limited hearing capacity for loud, low-frequency sounds, as they lacked a tympanic middle ear. The first terrestrial vertebrates had an auditory papilla that rested on solid tissue and was covered by an otolith, a small mineral structure present in the inner ear of certain animals, notably fish and reptiles, responsible for detecting movement and balance, and composed of calcium carbonate crystals. In amphibians, the otolith membrane was replaced by a tectorial lining that contained no otoliths. In all subsequent lineages, the basilar membrane appeared, freely suspended. In other lines, the basilar membrane showed some adaptation, with moderate frequency selectivity. Around 100 million years after the separation of the tetrapod lineages during the Triassic period, at least 5 independent developments of tympanic middle ears were observed. These developments functioned as essential impedance matching mechanisms for the efficient detection of airborne sounds. The absence of an effective middle ear prevents the detection of high-frequency sounds. This gave rise to parallel evolutions, leading to major differences in the auditory systems of tetrapod vertebrates between different clades, while ultimately fulfilling the same function: detecting, transmitting, transducing, and encoding airborne sounds. Paleontological, morphological, and developmental evidence suggests that lepidosaurs, such as lizards and snakes, as well as archosaurs, which include birds and crocodiles, independently developed a middle ear, composed of a single ossicle. Firstly, in birds and crocodilians, the structure of the auditory organ shows marked similarities, such as the presence and distribution of similar cell types. It is therefore reasonable to assume that this structural configuration was reached or initiated at the latest around the separation of their ancestral lineages some 160 million years ago. Similar reasoning applies to lepidosaurs. In Tuataris, which separated early from other lepidosaur lineages, the cellular configuration of the auditory papilla is similar than in lizards. In Tuataras, as in turtles, there is only one type of cylated cell, and all cylated cells have the same orientation. Thus, the more complex structure typically seen in lizards seems to have arisen after the Tuatara lineage separated from the squamates. However, it is not known whether lepidosaurs, including the Tuataris, developed a tympanic middle ear all at once. Finally, in mammals, the characteristic structure of the auditory organ, known as the organ of Corti, is essentially found in all recent groups of mammals, although there are marked differences between monotremes and therian mammals. We can therefore assume that the organ of Corti appeared before these lineages split, between 220 and 150 million years ago. This comparative analysis suggests that the evolution of the tympanic middle ear in each lineage had significant implications for the later development of the auditory organs of the inner ear. The reduction in bone size and the emergence of new configurations to form the tympanic middle ear probably improved not only auditory transmission in general, but especially that of higher frequencies. This new feature undoubtedly exerted additional selection pressures on the auditory organ to enable optimal transduction of information and its transmission to the brain. Over time, the mammalian cochlea evolved specifically in different subgroups. Over the course of several subsequent geological periods, mammals diverged considerably, and although the basic structure of their cochlea was relatively constant, differences in length and various specializations emerged. Not only did cochlea develop huge differences in length, they also developed specific specializations in groups pursuing different strategies for locating prey, using ultrasonic frequencies in the air, such as bats, and high ultrasonic frequencies in the water, such as toothed whales. For example, some bat species have developed cochlear regions where the length of the organ of corti, dedicated to specific narrow frequency ranges, is considerably enlarged. These adaptations have enabled bats to better detect and localize sounds in their environment, improving their hunting and navigational abilities. On the other hand, there is no reason to expect mammalian auditory organs to be necessarily superior or more sophisticated than those of other groups. Each present-day vertebrate group has developed an appropriate auditory solution according to the evolutionary pressures dictated by the environment, predator-prey relationships, and intraspecific communication needs. Acute sensitivity and precision in frequency resolution, characteristics commonly associated with mammalian auditory organs, have evolved independently in all groups of modern terrestrial vertebrates, in response to their own specific evolution and adaptation. Indeed, some species of lizard possess auditory nerve fibers tuned to some of the finest frequencies of any vertebrate, including most mammals. Birds of prey, particularly owls such as the barn owl, rely almost exclusively on hearing to locate their prey, so auditory sensitivity exceeds that of most mammalian species. Echolocating mammals such as bats and dolphins exhibit a wide range of hearing frequencies, a characteristic almost equaled by certain species of frog, which use ultrasonic calls to communicate. In humans, although generation times are 10 times longer than in fish, the issue is not so much the creation of hundreds of new species, but rather the adaptation of a sensory organ to optimize the processing of communication signals vital for survival and hence reproduction. Whether the evolution of the human cochlea paralleled the development of language is a fascinating question that could be explored by future research. All living cells are irritable to certain chemicals, and this predisposition of cells to be perturbed by chemicals is likely to have led to the eventual evolution of specific receptor proteins to detect chemical signals, and ultimately to specific chemosensory organs and systems such as the sense of smell. Olfactory capacity, used for chemical communication, is widespread in the majority of living beings belonging to the animal kingdom, enabling the perception and identification of chemical signals. The sensitivity and chemical range of animals' olfactory systems are remarkable, giving them the ability to detect and distinguish between thousands of different odor molecules. In the animal world, olfaction has evolved to enable the detection of signals from the chemical environment, which contains decisive information on the direction to follow, food sources, the right time to reproduce, as well as stimuli to consider pleasant or dangerous. An animal's reaction to chemical signals is the result of a combination of mechanisms acquired during its lifetime and innate predispositions resulting from the cumulative impact of individual experience and evolution through the generations on the olfactory nervous system. Evolution has shaped the olfactory system in response to two challenges: the need to explore a wide range of chemical information and the specific sensory challenges faced by each animal species. Generally speaking, vertebrates develop an olfactory system that detects odorants and pheromones through their interaction with cell surface receptors specialized on olfactory sensory neurons and organized in dramatic and subtle ways, enabling adaptations in response to major changes in environment and lifestyle, such as the transition from ocean to land and the predominant development of vision. In terrestrial vertebrates and insects, we often think of olfaction as a chemosensory modality dedicated to the detection of low concentrations of airborne volatile chemicals. Yet aquatic fish and crustaceans, while not encountering airborne volatile odorants, possess sensory systems anatomically similar to the olfactory systems of terrestrial animals. For these aquatic animals, odors are sapid molecules in solution. Olfaction is therefore not necessarily the detection of volatile molecules in the air. In this context, it has been suggested that during the transition between aquatic and terrestrial environments, new demands emerge for chemosensory systems linked to odor detection. This happened in a remarkable way, as olfactory stimuli evolved from hydrophilic to predominantly hydrophobic and volatile compounds. This transition granted terrestrial vertebrates an evolutionary option to detect volatile water-insoluble odorants. As a result, it is thought that the complex biosynthetic pathways required to produce volatile compounds, as well as the highly complex combinatorial coding that characterizes olfactory perceptions, driven by olfactory receptors, emerged suddenly in just a few 100,000 years. Comparative genomic and phylogenetic studies argue that chemosensory receptors were conserved across aquatic and terrestrial taxa, and that mammalian-like olfactory receptor genes evolved in marine environments before the first vertebrates appeared. These changes in olfactory receptors at protein level, altering their interaction with odorant molecules, play a key role in the evolution of olfactory perception. However, researchers have discovered that this adaptive evolution of olfactory perception can also be influenced by co-expressed olfactory receptors undergoing changes in copy number and protein sequence. Selection also acts on individual olfactory receptor families or subfamilies, favoring specialized adaptation or facilitating chemical communication between members of the same species. Finally, genetic variation among the thousands of unique olfactory receptor genes can lead to behavioral modifications in response to specific chemical signals. Thus, the combination of these mechanisms enables vertebrates to adapt in precise and complex ways to their changing chemical environment. An animal's genome evolves in such a way as to be closely linked to the specific tasks resolved by its nervous system, enabling it to detect odors and respond appropriately to promote survival and reproduction. Recent studies have revealed the presence of ancestral versions of synaptic genes involved in fundamental neuronal communication in various organisms without a nervous system, suggesting a progressive evolution of olfaction. In parallel, studies have clarified that the olfactory capacities of multicellular organisms, as well as chemosensory learning abilities, evolve from life forms not equipped with complex neural networks. In particular, prokaryotes have been proposed as model systems for understanding the evolution of olfactory systems in higher organisms. In fact, bacteria respond to volatile chemical gradients, but also use contact-dependent signaling. In addition, a unicellular but multinucleate eukaryotic organism, Viscerum polycephalum, has been shown to be capable of chemosensory detection. Studies have shown that olfactory receptor cells are primary bipolar neurons with a similar morphology between species. They have filamentous dendrites to increase the surface area for stimulus capture, and an axon that extends to the central nervous system. Dendritic processes are mainly of ciliary origin in most animals. Although the overall morphology is conserved, the fine details of olfactory receptor cell morphology can be adaptive, i.e., they can vary in a habitat-dependent rather than species-dependent manner. For example, the olfactory receptor cells of land-dwelling crustaceans, such as the giant thief crab, have a morphology closer to that of insects than to that of marine crustaceans. Crustaceans share a common ancestor with insects, and as they lack olfactory receptors, it has been proposed that these receptors evolved when prehistoric insects left the sea to live on land. According to this idea, olfactory receptors evolved because these ancestors must have been able to detect odorous molecules floating in the air, rather than dissolved in water. But a recent discovery contradicts this hypothesis. The discovery of a new type of olfactory receptor, ionotropic receptors, IR, has changed the order of things and opened up new avenues of inquiry into the evolution of olfactory receptors throughout the history of life. These new olfactory receptors are present in crustaceans, in which no classical olfactory receptor had been found until now. Since crustaceans were the first to appear in the history of life, before insects and before vertebrates, we can assume that these IR were inherited long before the appearance of the first insects. During development, the olfactory system takes shape from the olfactory placodes, specialized regions of the anterior ectoderm that share cellular and molecular characteristics with other placodes involved in the development of various cranial senses. Certain cell types of the olfactory system preceded the appearance of vertebrates, as did certain mechanisms involved in placode formation. It is likely that these two elements were already interconnected in the common ancestor of vertebrates and tunicates. In vertebrate ancestors, this system evolved into a more complex organ system, integrating additional tissues and specific morphogenetic processes to develop distinct olfactory components. Subsequently, the ancestral placode divided to give rise to the paired olfactory organs characteristic of most modern vertebrates. Thus, a major olfactory adaptation occurred along the amphibian and terrestrial branches of the vertebrate tree, resulting in the formation of distinct neuroanatomical structures expressing more recently derived olfactory receptors. Aquatic vertebrates such as fish and amphibians have an olfactory system similar to that of terrestrial vertebrates, but specialize for the detection of chemicals present in water rather than air. Insects, on the other hand, have a well-defined system in their antennae for detecting airborne chemicals, generally referred to as the olfactory system. However, this system is evolving convergently, independently of vertebrates, to perform this function. Unlike fish, amphibians have developed a separate, vomeronasal organ, which specifically detects attractive sex pheromones specific to amphibians. In addition, they possess an air-filled main olfactory epithelium, which expresses a vast repertoire of novel Class II olfactory receptors that have acquired a role in innate reproductive behaviors. These observations suggest that odor interpretation depends not only on the odorant molecules detected, or ligands, but also on the developmental identities and specialized sensory pathways of the cells that detect these odors. Recent evidence shows that individual mammalian olfactory receptor cells can be activated by several different odorants, and that individual odorants activate several receptor cells expressing different receptor proteins. This combinatorial coding is also present in insects and fish, suggesting that it has been conserved in the evolution of olfaction. Consequently, there are striking similarities between species in the organization of the olfactory pathway, from the nature of olfactory receptor proteins to the organization of the olfactory central nervous system. Although there is a striking evolutionary convergence towards a conserved organization of signaling pathways in vertebrate and invertebrate olfactory systems, the receptor gene families involved have evolved independently. These common organizational features span a wide phylogenetic range of animals. Such conservation also implies that there is an optimal solution to the problem of odor detection and discrimination. Either this solution evolved relatively early and was then retained in evolution, or more likely, animals convergently developed identical or similar solutions to the odor detection and recognition problem. The sense of taste is a specialized chemosensory system, one of the main mechanisms used by animals to assess the nutritional quality of food, helping them to make decisions about consuming beneficial foods while avoiding harmful substances in their daily lives. Unlike touch, vision, hearing, or olfaction, which are called upon in different behavioral situations, the sense of taste has evolved to play a primordial role in food regulation and motivation. Taste systems detect essential nutrients, as well as harmful substances in food, and trigger innate behaviors that lead to the acceptance or rejection of potential food sources. Most vertebrates can perceive 5 basic taste modalities: bitter, umami, sweet, salty, and acidic. All vertebrates have some ability to avoid harmful chemicals and seek out nutrients. It is thought that each sense of taste has evolved to detect and motivate the consumption of essential nutrients, and also to detect and avoid potential poisons. It is widely accepted that sweet taste evolved in plant-eating animals to detect energy-rich simple sugars, such as glucose, fructose, and sucrose. Bitter taste, on the other hand, probably functions to ensure that an animal avoids poisons. Indeed, most poisons are bitter, and most bitter substances are harmful, although this relationship is not perfect. The salty taste allows the detection of sodium, an absolutely essential mineral. When certain species of animal lack sodium, and this usually occurs in herbivorous animals, a powerful appetite for salty taste is aroused. And for many species, salt is consumed even when there is no apparent need. As for the sour taste, many have suggested that it is involved in detecting fruit ripeness. Finally, the fifth basic taste, umami, probably serves to signal amino acids and proteins. By comparing animals from different branches of the evolutionary tree, researchers have deduced that the sense of taste probably evolved over 500 million years ago. This would have occurred before the divergence of terrestrial vertebrates, bony fish, sharks, and lampreys, when their common ancestor, a primitive fish, developed a new type of cell. These chemosensory taste receptor cells, located in taste buds, are able to detect chemical stimuli and send signals to the brain via the taste nerves to generate taste sensations. Taste buds are the gustatory end organs in vertebrates ranging from lampreys to mammals. These end organs respond to a variety of sapid chemicals and transmit signals to afferent nerve fibers from 3 cranial ganglia: facial, glossopharyngeal, and vagus. Over time, taste buds have undergone several modifications to adapt to the dietary needs of different animal species, which explains their varied location in these animals. Not only are they found in the mouth and pharynx, but they can also be widely distributed across the surface of their bodies. Evidence suggests that umami receptors were the first to develop. Researchers have reported the discovery of genes similar to those encoding the receptors used by humans to detect the amino acid glutamate in the genome of the elephant shark, a species that split off from other fish 400 million years ago. Sharks lack bitter taste receptors, suggesting that these genes evolved more recently. Ancient fish were probably the first vertebrate animals capable of detecting sour taste. It is likely that these fish did not taste food with their mouths, but rather could detect acidity in the ocean using the outside of their bodies. Variations in the carbon dioxide dissolved in water can create different levels of acidity, which can be dangerous for fish. So the ability to sense acidity would have been essential for their survival. Bitter taste is perhaps the taste that most intrigues evolutionary biologists. It's the Darwin's finch of taste, engineered and customized to suit a species' ecological niche. Humans have 24 or 25 different bitter receptors, depending on the individual, each recognizing unique combinations of chemicals. Toxic bitter compounds come in all shapes and sizes, so it makes sense that the receptors that recognize them are diverse. The evolution of bitter taste, therefore, also involves distinguishing between different chemicals. The diets of humans and their ancestors are mainly based on plants, which are an abundant and accessible source of nutrition. However, plants have developed physical, reproductive, and chemical defenses to protect themselves from herbivores. Herbivores must therefore contend with various obstacles such as thorns, shells, and symbioses with defending animals. Herbivores have developed various strategies to overcome the toxic defense mechanisms of plants. Among these, a particularly effective and widespread method in vertebrate animals is the detection of bitter flavors. It has long been widely recognized that many plant toxins are perceived as bitter, creating a powerful mechanism for minimizing exposure to these harmful substances. By signaling the presence of toxins in foods before they are fully consumed, bitter taste detection helps avoid their ingestion. This ability plays an essential role in the survival of herbivores, helping them to identify and avoid potentially dangerous plants. The ubiquity of bitter perception in vertebrates and its complex role in humans have raised questions about its evolution. One of the most remarkable revelations of vertebrate comparisons is that bitter taste receptors are a relatively recent evolutionary innovation. While they are present in bony fish, lobe-finned fish, and tetrapods, they are not found in cartilaginous fish. Thus, bitter taste receptors must have first appeared in an aquatic species around 430 million years ago. This makes them the most recent of the chemosensory receptor families in vertebrates. The origins of the receptors involved in taste perception in animals are the subject of much speculation. Similarities between the evolution of vascular plants and the Cambrian explosion in animals suggest that the origin of these receptors may be linked to an important transition in plant to herbivore interactions. However, there is also evidence that these receptors play other roles, such as detecting nutrients in the gut, suggesting that their function as taste receptors may have emerged later. The bitter taste receptor family has diversified within and between taxa, with repertoires of bitter taste receptor genes varying in size across species genomes. Studies show that selective pressures on taste perception represent the most obvious potential driver of diversification of these genes. Fish have different numbers of genes, ranging from 1 in Amazon molly to 74 in coelacanth, but most fish have an intermediate number of genes. Variation in the number of bitter taste receptor genes occurs in different groups of animals, such as fish, amphibians, and saurians. Caecilian amphibians, which include limbless amphibians, have the lowest number of genes, while anarans, which include frogs and toads, have the highest number among all recorded vertebrates, probably reflecting their adaptation to variable lifestyles and environments. In fact, most frogs and toads inhabit both aquatic and terrestrial niches, which necessarily contain a greater variety of toxic substances. Consequently, their ecological needs should encompass very different requirements for their gustatory systems. For example, aquatic factors such as pH have been shown to influence the divergence of taste receptor genes. Most frogs feed on worms, insects, and other small arthropods, which contain more potentially toxic substances. It is known that insects often sequester toxic compounds from plants for their own defense, suggesting that anarans may benefit from the ability to detect dangerous prey. While caecilians are animals adapted to life underground, living in soil layers rich in organic matter, they build tunnels to get around. All caecilians are carnivores, feeding on earthworms, plathelminths, arthropods, frog eggs, and tadpoles. Compared with frogs, caecilians are less sensitive to bitter taste, as animal tissues contain fewer toxic substances. Consequently, we hypothesize that the lifestyle, ecological, and dietary complexity of amphibians raise their evolutionary pressure for a wide variety of taste receptor genes. Saurians, which include crocodiles, lizards, snakes, and birds, also show variation in the number of these genes. Snakes have low numbers, while crocodiles, turtles, and lizards have higher numbers, similar to many fish. Repertoires of bitter taste receptor genes in birds and mammals show great diversity. Penguins and cetaceans have few or no functional genes, while birds such as zebrafinch and white-throated sparrow have several. Primates, including man's closest relatives, have an average number of genes, with the gibbon having the fewest and the chimpanzee the most. Although toxins are a threat to all herbivorous animals, not all species are equally exposed. This variability is reflected in the size of genetic repertoires, which is related to the proportion of plant-based diets for each species. This trend is particularly marked with herbivores having larger repertoires than carnivores. The most extreme example of this variation is found in obligate carnivores such as whales, penguins, and pinnipeds, which swallow their food without chewing, resulting in a total absence of certain genes linked to toxic detection. Around 7 to 8 million years ago, humans had one last common ancestor with other great apes. If we look at the current eating habits of great apes, we can deduce the diet of our common ancestor. It seems that this species was omnivorous, feeding mainly on tropical fruits, and also included leaves and insects in its diet. Chimpanzees, our closest relatives, rely heavily on fruit for their caloric intake, with a small proportion of their diet coming from animals such as other apes and insects. Between 4.4 and 2.3 million years ago, human ancestors left the forest for the savanna and other ecosystems, and their food repertoire expanded considerably. Thus, over time, early hominids gradually abandoned the eating habits of forest monkeys and increased their ability to obtain a variety of nutrients in order to adopt a more diversified outdoor diet. Despite this dietary evolution, our need for fruit and our attraction to sugars and acids, shared with other great apes, remained intact. From a nutritional point of view, fruit is attractive because of its sugar content, which provides intrinsic satisfaction, as well as vitamin C, which is essential for life support in hominoids. The question of why humans are sensitive to acidic flavors and even inclined to prefer acidity has sparked debate in the scientific community. Signals from acidic substances do not provide much nutritional value, with a notable exception of vitamin C. However, this exception is significant because, unlike most mammals, monkeys and apes are unable to synthesize vitamin C due to the loss of a functional version of the gene encoding gluconolactone oxidase. The common ancestor of arthropoids that suffered this enzymatic loss probably had to rely on a sufficiently rich source of ascorbic acid from fruits and other plants for the enzyme to become useless. It is plausible that the acidic taste played a necessary role as a guide to identifying vitamin C-rich fruit. In addition, the combination of acids with sugars could also have been used to assess the degree of ripeness of the fruit, using mixtures of sweet and sour flavors. Thus, acids are not stimuli for which we have evolved to react in isolation, but rather elements we have experienced in the context of the sugars present in fruit. As a result, sweet and sour flavors are perceived as synergistic in the fruit aroma palate. In addition, acid and sour taste act as indicators of fermentation processes which human beings around the world seek out and consume. Wild apes depend mainly on forest plants, especially young leaves, for most of their daily protein intake. Nevertheless, early hominids and modern humans have shown a preference for consuming slightly more digestible protein sources, such as meat. Although umami taste is not immediately apparent in fresh meats, it develops significantly in aged or cooked meats. Consequently, it seems that chimpanzees do not possess a gustatory system specifically dedicated to detecting the taste of glutamate or ribonucleotides, even though they can perceive these stimuli. The characteristic umami taste of hydrolyzed protein is mainly carried by glutamate and ribonucleotides. Humans have developed a preference for glutamate, ribonucleotides, and umami taste, probably as signals indicating more easily digestible proteins in lightly aged or cooked meats. Indeed, there is ample fossil evidence to suggest that culinary practices predate the advent of modern man. Humans the world over take advantage of a wide variety of plant and animal products that undergo fermentation. Our strong attraction to the taste of free amino acids and ribonucleotides may stem from a tendency to consume fermented foods, including lightly aged and/or cooked meats. This category of food would bring various benefits to the survival of our species. Fermentation facilitates access to macro and micronutrients while providing probiotic bacteria. Although it has been assumed that the pleasant taste of glutamate and ribonucleotides acts as an indicator of protein, many protein-rich foods are not particularly tasty or umami when fresh. It's the process of fermenting or aging these foods that releases the umami, savory taste of protein. Thus, our attraction to amino acids, particularly glutamate, as well as salty taste, may have emerged from our desire for fermented foods, taking advantage of the nutritional improvements and benefits of probiotic bacteria for our species. Humans stand out from other mammals thanks to a unique phenomenon, a large-scale polymorphism of the salivary amylase gene. This change in our genetics could be the result of our past history linked to the consumption of starch-rich tubers, and could have occurred around 100,000 to 200,000 years ago. The number of copies of the salivary amylase gene present in our genome directly influences the production of amylase in our saliva. This enzyme in high quantities and efficiency has the ability to rapidly transform a cooked starch, such as a thick pudding, from a semi-solid to a liquid state in a matter of moments in our mouths. The resulting sugar concentrations can produce sufficient substances such as maltose, which activate brain regions linked to taste and reward. In short, it seems that the human mouth harbors a taste receptor that responds to glucose polymers without necessarily inducing a conscious taste sensation. This receptor can be stimulated by the products of starch decomposition in the mouth, especially in the presence of high levels of salivary amylase. Human beings have a natural tendency to seek out the taste of salt in food. We find the taste of moderate concentrations of salt very appealing, as do other omnivores. But high levels of salt are considered unpalatable to us, as they disrupt the osmotic balance of our body fluids. Salt is a widely used ingredient in food preparation throughout the world, and individuals from different cultures consume roughly the same amount of salt every day. This similarity in salt consumption across cultures suggests that our salt intake is regulated by intrinsic biological factors. A carnivorous animal absorbs salt at every meal, while a herbivore can easily run out of sodium, triggering a desire for salt, which translates into a search for natural licks. An omnivore's exposure to dietary salt would therefore logically be intermediate between that of carnivores and herbivores. It should be noted, however, that humans lose salt through perspiration, which may explain why we have a slightly higher salt intake preference than other omnivores. Since our earliest prehistoric ancestors, the sense of touch has been central to our survival and reproduction. What we touch can have a direct impact on our ability to survive and reproduce. It may be a predator, prey, or parasite, a parent, lover or child, shelter or danger, a tool or food source, the ground beneath our feet, or the branch we use to swing. It's also the first sense to develop, with some fetuses responding to touch in the womb after just 8 weeks. Evolution has created a tactile system made up of different sensors and receptors, which together detect the qualities of what touches our body and what our body touches, such as weight, temperature, and texture, so that we can deduce useful information. Many animals, including some coelantherates, annelid worms, insects, and many other arthropods, birds, and mammals, have hairs or capillary projections that are richly supplied with nerves and serve to indicate to the animal that it is in contact with an object. These hairs may have been specially modified, for example, vibrissae or whiskers in certain areas of the body, such as the face or toes, to provide more sensitive discrimination between stimuli. A second type of tactile receptor, the subcutaneous receptor, is found in the skin. Receptors of this type are found in almost all animals and may consist of free nerve endings or complex corpuscles. Unicellular organisms already possess the capacity for directional detection of tactile stimuli, as can be seen in ciliates such as paramecia, which move away from the stimulus source following mechanical disruption of their outer membrane. Sponges, the simplest multicellular animals, do not possess neurons, but they do have the capacity to respond to variations in water flow and pressure in response to the movement of their immobile cilia. However, the evolution of neural conduction has led to a radical change in the ability to respond rapidly and flexibly to tactile stimuli. Cnidarians such as jellyfish and hydra, despite their relatively simple nervous systems, can exhibit coordinated patterns of motor response to sensory stimuli, and many have a rich capacity to respond to touch. For example, hydra nematocytes are structures that provide a good model for understanding the mechanoreceptors of more complex invertebrates. In jellyfish, groups of hair cells called tactile combs regulate complex behaviors, including escape, feeding, and locomotion. The advantages of sensitivity to mechanical stimuli provided by hair-like structures may have encouraged their convergent evolution in several animal lineages. For example, the ciliated cells of jellyfish appear to be sufficiently different from those of vertebrates that a common origin for both is unlikely. Studies into the molecular basis of mechanosensation in different classes of animals also suggest that the cellular mechanisms required for tactile detection may have evolved several times. All animals with a central nervous system respond to touch. The nematode, Sanor habditus elegans, is an important model organism in neuroscience. It is the first animal system in which the entire neural network involved in tactile behavior has been identified. This worm has mechanosensory cells located in the integument beneath the cuticle, linked to 6 sensory neurons, 10 interneurons, and 69 motor neurons. In larger, probably more advanced animals, touch-induced behavior becomes more versatile and complex. The leech has developed a central nervous system with a succession of ganglia for each segment. Each ganglion houses 3 groups of mechanosensory neurons, all of which respond to mechanical stimulation of the body, but with different response thresholds. Tactile cells are the most reactive and respond to the gentle touch of the body wall. Pressure cells respond to more intense tactile stimuli, while nociceptive cells are sensitive to very powerful and potentially harmful stimuli. In arthropods, mechanoreceptors are located in strategic areas of the body where movement is most likely to occur. In proprioception, these are the joints, muscles, and tendons. In contact, they are the surface areas where contact with external objects is most likely. Arthropods exploit these strategically important locations in 2 ways. On the one hand, they use tufts of hairs called capillary fields, or capillary plates, to detect movement between adjacent body segments through proprioception. On the other hand, to detect contact, they use similar hairs but located in different parts of the body, with a particularly high concentration where contact is most likely and/or most important to detect. Spiders and insects also use hair sensilla for proprioception and consequently for active touch detection, where the active movement of a limb must be monitored. Among today's arthropods, only myriapods, crustaceans, and insects have specialized touch sensory structures, the so-called antennae. These sensory organs are equipped with a particularly large number of sensillae, displaying a density far greater than that found on most other body regions. Crustaceans have 2 pairs of antennae, while insects have only 1 pair. The generally accepted hypothesis is that over the course of evolution, insects are likely to have lost their 2nd pair of antennae, meaning that the insect antenna shares a common evolutionary origin with that of crustaceans. Fish and amphibians have mechanoreceptive organs called neuromasts, which are arranged in rows on their body, head, and tail. These neuromasts may be present autonomously on their skin or inside fluid-filled channels. The main sensory component of each neuromast is a group of hair cells that respond to hydrodynamic stimuli caused by water movement or variations in water pressure. In this way, The lateral lines provide a sophisticated sensory system for detecting the distant origins of water movement, such as predator or prey behavior, water currents, and the topographical features of the underwater environment. Mammalian skin evolved from the integument of earlier vertebrates in a complex evolutionary process that is still only partially identified. What is clear is that the first mammals, or perhaps their reptilian, therapsid ancestors, redeveloped hair as an adaptation of the outer epidermal layers of their integument. While dense hair clearly has a thermoregulatory function, the evolution of hair in mammals cannot be attributed solely to the goal of regulating body temperature. On the contrary, it is very likely that the first hairs were primarily used for tactile functions, and their subsequent evolution gradually gave them a secondary role as insulators. The main sensory function of hairs persists in vibrissae, also known as tactile hairs, present in all therian mammals, such as marsupials and placentals, with the exception of human beings. In cases such as the naked mole rat, a species that has lost its entire coat, tactile hairs have been maintained throughout the body because of their importance to the sense of touch. Many mammals have evolved to have areas of hairless skin, also known as glabrous skin. In humans, these areas include the skin of the lips, hands, fingertips, and soles of the feet. These are the parts of the body that play a key role in physical interactions with the outside world, and where tactile precision is particularly important. So it's not surprising that hairless skin is characterized by a high density of mechanosensory receptors that are extremely sensitive to delicate touch, which could contribute to the effective or social tactile capacity that could be specific to mammals. Only mammalian species have elongated facial vibrissae, also known as vibrassae, which extend outwards and forwards from the animal's snout. They form a tactile sensory network that envelops the animal's head. Species without vibrassae, or in which they were less apparent, were generally the most evolved representatives of their order. Consequently, having facial vibrassae was an ancestral characteristic of mammals. Many terrestrial mammals, especially those that are nocturnal, live in low-light environments or climb trees, and have evolved highly specialized tactile sensing systems via vibrassae. Research has focused on a variety of these animals, including rodents such as the rat, mouse, and golden hamster, as well as insectivores such as the Etruscan shrew and the Brazilian short-tailed marsupial possum. In some seal species, vibrassae have adapted to detect disturbances left in the water by fish. By measuring these hydrodynamic flow fields, these animals are able to track and capture fast-moving prey in total darkness. Deep-sea foraging pinnipeds, such as the bearded seal and walrus, have evolved a highly specific vibrissal adaptation designed to optimize their efficient search for shellfish and other invertebrates in the muddy layers of the seabed. The abundant, dense, and mobile vibrassae of these animals combine to form a kind of sensitive, tactile rate that enables them to detect and ingest their prey at exceptionally fast speeds. Manatees have vibrassae distributed throughout their bodies to compensate for the limited visual information available in their natural environment. The manatee's ability to move efficiently through rough waters suggests an overall body ability to perceive hydrodynamic signals to the sensitivity of its vibrissae, which has interesting parallels with the lateral line of fish. These adaptations underline the importance of vibrissae touch detection in the ecology of many aquatic mammals. Human fetuses begin to sense their environment through touch as early as the 8th week of gestation, contributing to their body awareness. After birth, the mouth evolves to become the primary means of discovering surrounding objects. But this haptic focus shifts to the hands as we grow older. It's not surprising that humans lost their facial whiskers over the course of evolution, as our hands took over as tools for tactile exploration of the immediate environment, while also enabling us to manipulate with skill and precision. Unicellular and multicellular organisms have developed different strategies to ensure their survival and communication with their environment. These strategies include molecular, anatomical, and physiological systems that enable them to detect and respond to chemical signals from other cells or organisms. These communication mechanisms are the result of organisms' ongoing adaptation to environmental factors and biotic interactions. Over time, organisms have developed adapted receptors that enable them to detect and respond to specific signals.